Good afternoon. Welcome back to Amplify the Signal. I hope you enjoyed your lunch break. We have a request from our online watchers that we use the microphones for questions. So we have two nice volunteers, Jeff Hoover and Katie Lee, will be helping with that. So when it's time for questions, please do use the microphone and hold it right up close so we can all hear the questions. Thank you. Well, we have a lot of science writers here, so you know how easy it is to succumb to temptation. The temptation of cliches and puns, especially when the topic is bees. May Berenbaum addresses that right up front in the title of this session, The Buzz About Bees in the News and Other Accompanying Bee-Labored Puns. I might say that her book about honey, Honey, I'm Homemade, is available for sale, and she'll be part of the book signing later. And the first 30 people to buy that book get a 3-ounce sample of honey from the University of Illinois bee facility. So, May is a professor and head of the Department of Entomology at the University of Illinois. She studies interactions between plants and insects in natural and agricultural ecosystems. Her work has garnered considerable recognition, including the National Academy of Sciences membership and the 2014 National Medal of Science. She is the editor-in-chief of the Proceedings of the National Academy of Sciences. Devoted to fostering scientific literacy, she has written articles and books about insects for the general public. She has been interviewed by media hundreds of times and has testified before Congress on pollinator decline. Always a leader in public engagement, she founded the University of Illinois Insect Fear Film Festival, a festival that indulges in Hollywood's etymological excesses. Please welcome Professor May Berenbaum.
Thank you. Hi, am I on? Can you hear? Yes. Well, thank you so much for inviting me to be part of this. Science communication is a passion of mine. This is an unusual presentation for me. I'm more accustomed to talking straight science. This is a little more personal. It's distilled down. Well, it covers about a decade worth of personal revelations and insights into the power and perils of amplifying the signal. Apparently, if you over-amplify, you can distort the wavelength. I learned. With luck, it will fit into the 50-minute slot. All right. The alternate title for this talk could just as well have been My Life on the Bee List. And the reason for that is, well, it should become apparent. So a little bit of backstory. In August 1980, I deposited my thesis at Cornell. 2 weeks later, began as an assistant professor in the Department of Entomology right here at University of Illinois, Urbana-Champaign. One job my entire adult life. So I came here after working with Paul Feeney, testing the hypothesis of Ehrlich and Raven, which sort of modeled after the sort of Cold War dynamics of the time, envisioned that interactions between insects and the plants they eat is like an arms race, ever-escalating arms race, where plants develop defensive chemicals, insects develop counter defenses, and that goes on reciprocally for millions of years. I replaced Peter Price as the insect ecologist here, and brought my interests in plant-insect interactions to Illinois. My main focus, even now, has been on a particular plant family, the Apiaceae or carrot family, producing a particular class of plant defenses called foranocoumarins. These are light-activated chemicals that can cross-link strands of DNA and cause all kinds of problems to all kinds of living things, including a condition called photophytodermatitis, this unsightly rashes on painful, unsightly, oozing, blistery rashes due to the action of these defensive chemicals. So wild parsnip is sort of the classic weedy, noxious, apiaceous plant that is nonetheless consumed by herbivores that have overcome those defenses in a series of escalations of biochemistry and counter-biochemistry. Worked well here in Illinois because it's a roadside weed, lots of roadsides in Illinois, and because I'm afraid to drive, it was easy, got lost easily, and most of my sites were right by road signs with directions, so. I also brought with me an interest in cytochrome P450 monooxygenases. These are enzymes in a huge gene superfamily that have the main function in most aerobic organisms of breaking down foreign substances, xenobiotics, poisons, toxins, including phytochemicals and pesticides. So this is true. These were the defenses that, these were the counter defenses that insects have evolved to consume plant phytochemicals. I established many productive collaborations, including with Art Zongrel in entomology to understand the genetics of this coevolutionary process, and Mary Schuler in plant biology to gain some mastery of the biochemistry and molecular biology of these enzymes. So we could work on their heritability, enzyme activity, their upregulation of the genes, the structure of the enzyme. We could model the structure of the enzymes. So the question is, how on earth did I become, as one account put it, a bee expert? Not just a bee expert, a noted bee expert. And not just a noted bee expert, but the world's foremost bee entomologist, whatever that is. Moreover, I knew personally that not only I am not the world's foremost bee entomologist, I'm not even the campus's foremost bee entomologist. That honor really belongs to Gene Robinson. So in 2005, after 18 years of working on caterpillar cytochrome P450s, these are swallowtails, Gene Robinson, who headed up the Honeybee Genome Consortium, generously invited our group to annotate the cytochrome P450 genes in the Honeybee Genome Project. As it happened, I had a student, Reed Johnson, who'd just come to work in my lab after completing a master's thesis at Wake Forest on caterpillars and plants, because that's what my lab was known for. But he had worked as a summer intern with a bee biologist in Montana, Jerry Bromenschenk, and he took on the P450 annotation project as his own, and in the process, he contributed to revealing one of the more interesting findings of the Genome Project, and that's the fact that honeybees are depauperate in detoxification genes. They have fewer, a half to a third the number of detoxification genes encoding enzymes that break down poisons. So that was a surprising and intriguing finding. And we looked in the literature, and no one had done any work on honeybee detoxification of pesticides for at least 20 years. So we kind of changed our focus to figure out how, with such a reduced inventory, honeybees coped with natural and synthetic poisons. All right, so I was new to bees. My previous experience with bees between 1981 and 1984 was part of an ill-advised experiment in bucolic living that involved a horse, a pony mule, some chickens, geese, a banjo, and some bees. So when I discovered that actually geese eat bees, I did not know that. And until 2006, my only bee-related publications were 2 articles on honey chemistry because of my interest in phytochemistry, and nectar, of course, is a plant tissue loaded with phytochemicals, and an article on bees in heraldry in the American Bee Journal. So this is the background that I took into this project. But in the meantime, I was elected to the National Academy of Sciences based, of course, on my caterpillar work. In 2005, I stepped down as chair of the Board on Agriculture and Natural Resources in the National Research Council, the research arm of the National Academy, and I was asked to chair a study committee on the status of pollinators in North America. So the NRC study committees are sort of the gold standard. The National Academy was chartered to provide objective advice to the nation on scientific issues and policy. And actually, as chair, I approved this, the committee approved this study, and there were inklings in the greater world that there were pollinators who were in trouble. So for 18 months, we met, evaluated the literature, interviewed people, did surveys, and the like. In 2006, we released our report in October. So the committee was constituted to represent the full range of expertise involving pollinators. So I was chair of the committee. Jean Robinson was one of 3 people with legitimate honeybee expertise. So we were charged basically to figure out whether pollinators were declining, if so, why, what research and monitoring, what were the consequences, what research and monitoring would be needed, and how could those declines be stopped or reversed. So we released our report in October 2006, and one of our key findings was rather startling, that there's clear evidence that there's long-term population trends for honeybees in the US at the time were demonstrably in decline. And that, frankly, the other conclusion is we don't have enough data to assess pollinator status for most other species. But in the report, we also made a prediction. Basically, if those patterns of decline in Apis mellifera, the only reliable managed pollinator that is sort of semi-domesticated, if those were to continue at the rates we had seen in the past, there would be no commercial honeybee population by 2035. Well, timing is everything. October 2006 was also the first reports coming out of, quote, sudden alarming colony losses, a syndrome that later became known as colony collapse disorder. By February 2007, CCD, as it was known, had been reported in more than 20 states, significantly in California. And that picture there is of almond orchards. And every February in almond orchards, honeybees are imported. Half of America's honeybee stocks are loaded on flatbed trucks driven across the country, 2,000 miles or more, and delivered to these almond orchards to provide pollination services. 600,000 acres at the time, within a 2-week period, needed 1.25 million colonies of bees. And the almond growers and the beekeepers were worried that they would not have enough bees. Well, this was big news. This was big business news. On February 23rd, due to concerns about bee shortages in California almonds, the New York Times, the business section, picked up the story, and it turned out to be one of the most frequently downloaded stories in the entire month of February. So the New York Times reached out to me, because I chaired that committee, and asked if I would write an op-ed. It was kind of a thrilling experience for me. 3 days from the invitation to publication, 1 million potential readers, and they wrote a check for $400. So scientists are used to paying page charges. This was quite a nice turnaround. It clearly reached an audience. The upside of the New York Times op-ed is that 1 million people read it. That's also the downside, because then I got overwhelmed. The New York Times story led to an invitation on NPR's Talk of the Nation with Ira Flatow, during which I explained for the first, but by no means the last time, that it's a complicated situation. And this was a message that still has not resonated, but at the very earliest days, it was clear that this was complicated. Now, apparently, members of Congress read the New York Times, and the potential losses in this massive industry, $2.5 billion industry at the time, caught the attention of legislators in key states. California, where 80% of the world's almonds are grown. Florida has a massive honey industry. Georgia is the center of many queen breeding operations, so people can order queens for their bee colonies. The House Ag Committee Subcommittee on Horticulture and Organic Agriculture, that has responsibility for apiculture, decided to hold a hearing on colony collapse disorder at the end of March 2007. So, of course, there were the Congress people present. And word came to the Academy that they wanted a representative from the committee, the study committee. Who would it be? I kind of assumed it would be Jean. Jean, Swanland professor, director of the University of Illinois Honeybee Research Facility, 3 degrees in bee biology, 200 plus publications on bees, $10 million in grant funding to work on bees, and leader of the Honeybee Genome Sequencing Consortium. Okay. No papers, to my knowledge, on bees and heraldry. But Congress being Congress, I was the one they asked to testify. which was a wonderful opportunity to amplify the signal, as it were, of the findings of the National Research Council, NAS, study committee report. These, among all sort of scientific publications, these are regarded as the gold standard, even by members of Congress. So there was a great deal of interest in the details of the report. And it must have been a slow news day, because this room was packed with media. The money reporters from CNN, Bloomberg, ABC, CBS, NPR, PBS, and C-SPAN were all there covering this hearing for the House Agriculture Committee. And within days, the beepocalypse had been declared. There was sort of the national conversation turned. The expression fire hose was used earlier today about sort of this flood that can hit you. This was a fire hose, but I think at times it was silly string. So right on the heels of this pronouncement, an article was posted on a website, not refereed, not a journal, by a couple of physicists, not entomologists, who had done some experiments with digital enhanced cordless telecom communication. So a cordless telephone bases, which they put into beehives to see what the non-thermal influences might be on honeybees. Not cell phones. These were cordless phones. Yet, the Independent, a British newspaper, spun this into a story, our mobile phones wiping out our bees, which got such traction that for the first time in the memory of anyone at the USDA bee laboratory in Beltsville, the office of the vice president called, asking for a debriefing on the cell phone issue. So clearly, something had to be done. And a meeting was convened at the USDA laboratory in Beltsville of about 50 bee researchers, other interested parties who convened to prioritize colony collapse disorder research objectives. That was the goal of the two-day meeting. And we more or less agreed on the main hypotheses, which would set a research agenda. New class of pesticides, the neonicotinoids, novel pathogen or parasite. Honeybee apiculture has a long history of introductions of disease-causing organisms. Immune suppression relating to management practices, including schlepping them 2,000 miles across the country in the back of a flatbed. And declines in nutritional adequacy of the diet involving feeding them high fructose corn syrup for overwintering, or failing to provide them with suitable forage due to urbanization and development of natural areas and reduced plant diversity. What we de-emphasize were some of the other alternative hypotheses at the time, including, and not limited to, genetically modified corn pollen, cell phones, Wi-Fi, elevated CO2, elevated UVB, Osama bin Laden, automobile grills, solar maxima, jet chemical contrails, fluctuations in the Earth's magnetic field. This is for real. Soviet mind control experiments affecting the bees, alien abduction, and bee rapture, where bees are summoned up to heaven and leaving us behind. This meeting had no impact whatsoever on public speculation about the causes of colony collapse disorder. So again, a ream of articles appeared with favored hypotheses, including among environmental organizations, genetically modified organisms, and pesticides. And again, these were not seriously considered, well, genetically modified organisms, including corn, were not considered likely, in large part because bees really don't like corn pollen. It's a grass pollen. It's not particularly nutritious. It's not an insect-pollinated plant. Many other reasons, but that's neither here nor there. So my inbox started overflowing with interested citizens' suggestions. A biochemist from San Francisco Bay found some dead insects, some dead bees, and asked if I wanted them. And could he ship them to me? Very well-intentioned. Roseanne Barr had a radio show, and she invited me to be a guest. Apparently, she had a long-standing interest in bees. She dreamed the dream of bees my whole life, truly wished she'd been born orange and black. People thought I was crazy for the last 16 years to talk about the effect Chernobyl's accidents would quickly have on the food chain, but now the monthly extinction of different life forms seems to be an inescapable fact. Read about Chernobyl and the poisoning of the water, air, earth, and food chain. The bees are more evidence, I think. So that's what I had to talk to her about. Aries Keck, who runs a show called Earthbeat, asked me if I would be one of their guests. They have 3 segments per show, and would I be one of the guests on the segment? Sounded legitimate NPR. I did not know, however, that I would share that segment with Bill Fries, who at the time was with Friends of the Earth, who spent most of our time on air sort of berating me for not endorsing a study had been done by Hans-Hinrich Katz in Jena 2 years before there was any colony collapse disorder. This was a study examining genetically modified corn, BT corn, Bacillus thuringiensis corn, with an effect of honeybees with concentrated BT toxin 10 times the normal level. So raising these bees, and in the middle of his experiment, they all came down with a parasite, Nosema, a fungal parasite. Which normally you would just stop the experiment at that point because that was not part of the plan. He persisted and found a significantly stronger decline in the number of bees that occurred among the insects that had been fed this concentrated BT poison. And this was the evidence that Bill Fries cited to claim that I was being evasive in not indicting genetically modified corn as the cause for colony collapse disorder. And he pelted me with emails for months afterwards, although Aries Spear did friend me on Facebook. And then I got an email from a woman in Minnesota who works telepathically with companion animals and wanted to share with me her experience of telepathically communicating with bees to find out what the problem is. So she has a transcript here. Do you know what's going on with this bee disappearing thing? Bees. Yes, a worker bee goes out and does his work, and when he's ready to return to the hive, he turns toward the hive. At this point, the communication changed from words to a picture. I saw white lines in a grid pattern, parallel lines that converge in the distance, and parallel cross lines at a 90-degree angle. A grid. The background was dark, empty space. As the bee turns to home, he's on one of the lines that goes to the hive. He takes this line back to the hive. And I thought, something's wrong here. Maybe that transmission is garbled because, as far as modern science knows, it's the female bees, the foragers, that go out. Drones don't do anything except hang around, get fed by the workers, and mate with the queen and then die. So either her communication was scrambled or those drones were total posers. A helpful suggestion, again, in my inbox from an avid gardener worried about the situation, suggesting that maybe high-frequency active oral research program was responsible. So this is a high-powered energy-emitting device in Alaska that tampers with the magnetic ionosphere by boiling it and bending it 80 miles out into space, that's been steadily powering up. Its effects reach all of the US and halfway around the globe. And the National Colony Collapse Disorder Working Group, which I actually was not a part of, should not underestimate or play down the role that HAARP is playing, and she sent me a helpful internet link to a resource. Another hypothesis, at this time I was searching my name on Google News to see what was happening, and I was horrified to discover that I was behind another hypothesis, that Kevin Federline is stealing our honeybees. The weird thing about this story is it was partly true, and then there was ridiculous nonsense. So the phenomenon of disappearing bees first noticed late last year, that's true. Honeybees are used to pollinate $15 billion worth of fruits, nuts, and other crops, totally true. Prior to discovering honeybees in 1984, illegal Mexican labor was used at half the cost and half the time, though tougher labor immigration laws have made this method no longer feasible. So again, a paragraph that's reasonably true, followed by a reference to a professional wrestler named the Beekeeper, pointing out that if bees were dying of pesticide poison, they'd die at the hive, or from ingesting Alka-Seltzer, they would be tiny foaming corpses. And if they were abandoning the hive because of some threat such as zombies, they wouldn't leave without the queen. The main hypothesis is Kevin Federline is stealing the bees, said May Berenbaum, an insect ecologist at the University of Illinois. That's true. I mean, the insect ecologist at U of I part. Berenbaum has never liked K-Fed and blames him for turning former wife and pop sensation Britney Spears into the laughingstock of the entertainment world. Nationally, some 27 states have reported the disappearance. In each case, the bee disappearances coincided with a K-Fed concert, book signing, or paternity suit. And meanwhile, the more responsible press was having a field day with, like the title says, bee-labored puns. You have to admire the cleverness in some cases, although bee devils, farmers, beekeepers buzzing, what's bugging the bees buzz, comma, off, give bees a chance. Any John Lennon fans here? Bewildered, the flight of the honeybee, feeling the sting, a little historical reference to Roanoke, the lost colonies, a sticky situation, and befuddled, among others, many more. The problem is that beekeepers were seeing their livelihood at risk. This was not funny to them, and this seemed to be sort of a take on the news that was at variance with its actual impact on people's lives and on the ag economy. And then there's real fake news. You can always count on the Sun, came up with their own hypothesis that Africanized killer bees had mutated and started eating each other as cannibals, and that's why the bees were disappearing. So amidst all this, a photographer came down from Chicago to spend 3 hours taking photos of me in the breeze, on the quad, to choose a photo with my bra strap clearly visible to all 7 million readers. Right there. My parents were so proud. So in connection with National Pollinator Week, I was invited by Coalition for Agricultural Research to brief Hill staffers on the NAS report, and the next day to go to another hearing. This one was for the House Natural Resources Committee on the birds and the bees, how pollinators help maintain healthy ecosystems. And again, there are a collection of Congress people there and a panel of experts, including the Assistant Director of Fisheries and Habitat Conservation at Fish and Wildlife Service, Tom Lovejoy, very well-known conservationist, me, Danny Weaver, who president of the American Beekeeping Association and has a master's in biochemistry, a really remarkable individual, and Kevin Hackett, the program leader for bees and pollinators at USDA ARS. This infuriated beekeepers, in part because they thought that the conservation lobby was preempting the attention, sucking the air out of the room, and funneling away potential funding to solve their problem. So there are all sorts of bilious posts on various listservs, including this one that went on basically complaining, what's the subject of the hearing 3 months after we thought we were done explaining why CCD is serious? Hearing to explore nationwide decline of honeybees and other pollinators? Huh? What happened to CCD? Who spoke at this meeting? Then a sort of disparaging list of qualifications, including for me, number 3, May Berenbaum, an entomologist studies. What exactly? Not honeybees. Guy had a point, but I did chair that task force. After 3 months delay for no apparent reason, we honeybee guys are supposed to give a big round of applause to the inclusion of non-CCD related funding requests from people who have no interest in working on CCD at all. That was very frustrating for them. So frustrating, in fact, that he made the comparison that these people who are asking for support for research on other pollinators were basically like varroa mice parasitizing a honeybee, not coming along for the ride, but not being credited for helping the honeybee, just slowing it down. So I was basically being compared by beekeepers to a devastating blood-sucking parasite. It was heartwarming that one of my colleagues, who was an undergraduate in the Feeney lab when I was in graduate school, Dan Papage, came to my defense on this listserv. For the information of Mr. Fisher, I cannot personally imagine anyone I would rather have speaking to Congress on behalf of entomology and the Pollinator Protection Act, including the need for honeybee protection, than May Berenbaum, one of America's most prominent and highly regarded entomologists. What a nice thing to say. Moreover, Berenbaum has written a number of popular articles on bees, including a 1978 paper in American Bee Journal on bees as symbols in heraldry, which may be the only time that paper's ever been cited. So the USDA did a systematic collection of bees and shared them with the entire national research community in the hope of finding an answer. We were invited to participate, specifically to bring our expertise in pesticides and detoxification to bear on whether or not CCD had any connection to pesticides. So I was recruited to investigate honeybee toxicology based on our long history of working on caterpillar toxicology. In the meantime, other hypotheses were being explored, and Diana Cox-Foster and colleagues, using a viral metagenomics approach, demonstrated Israeli acute paralysis virus, not hitherto reported in North America, was associated with colony collapse disorder, marking the first time the popular press decided CCD mystery was solved. IAPV, described in Israel, 2004, it's a viral infection. 83% of CCD colonies were infected with this. And there was even a mechanism that in 2004, bees had been imported from Australia. Maybe that's where the IAPV had come from. But Australia may have had IAPV, but had no problem with CCD, and they were really annoyed by this. The claims in Science, that was a publication, Foster et al., very damaging for Australia, says a leading expert on honeybees in Australia. And to make things, I guess to clarify things, two enterprising USDA researchers, Jay Evans and Judy Chen, went into their freezers, pulled out historical samples of honeybees from 2001, looked for IAPV and found it there. So there's no way that IAPV could be the sole cause of CCD because it had been here all along. Okay, still searching the internet for CCD, I was horrified to find myself on this site that everyone else discounted the IAPV except this group, Israeli germ warfare virus blamed for honeybee decline. Sources in the U.S. Defense Department know where the virus originated, the Nest Tsiona Biological Institute, the main research facility for Israel's clandestine arsenal of chemical and biological weapons. This was a plot wiping out an enemy's food supply, level of pollination, enemy cannot feed its troops. The Israelis decided such a problem would hasten the end of hostilities by any superior aggressor. What is this site? What is SOS? It turns out it's synagogues of Satan. And I am afraid someday someone is going to audit my internet use and find out that I'm visiting this site. So, again, pursuing pathogens, pursuing pesticides. Pesticides were found in enormous, horrifying abundance in every part of beehives, every class of pesticides, including the most ubiquitous, and basically 100% of all wax was contaminated with the two mite-killing chemicals, acaricides, that beekeepers were putting in their hives to control the varroa mites. And in fact, this ubiquity of these two chemicals led us to test whether or not they interacted synergistically. Alone, they were thought to be safe for use with bees, but together, they were actually toxic to bees. So beekeepers were inadvertently introducing toxins lingering in the wax of the hive that could have ill effects. This got virtually no press coverage. Nor did this. This was a year-long study. It was a microarray analysis. So microarray is based on genomics. It's a way of visualizing expression of all 13,000, or I guess 10,000 plus genes in a honeybee genome, to see how they are regulated, which ones are turned on, which ones are turned off. And there should, in theory, be a footprint of the stressor expressed in the genome. If it's a pathogen, the defense immunity gene should be upregulated. If it's a pesticide, the pesticide detoxification genes, the P450s, should be upregulated. That's what we went to look for. Very difficult. We needed to find certifiably healthy CCD-free bees, which we did from the freezer. Different populations had CCD syndromes that differed slightly in their symptomatology. So it was a long, hard experiment. it yielded a very unexpected but illuminating result. We found no differential expression in immunity and detoxification genes, but in the microarray, we did find unusual ribosomal RNA fragments in the guts of the CCD beads. They may have been, we theorize, these are a consequence of repeated infections by RNA or ribosome attacking RNA viruses. And it was clear that honeybees are attacked by at least a dozen different RNA viruses, all of which can be transmitted by the varroa mites. So you break down the ribosome, that's the protein manufacturing center of the cell, then everything seems to cause CCD, because you can't make proteins for nutrition, for defense, for anything else. Seemed like a great story. Almost nobody wrote about it. I got some calls, but the minute I said ribosome, basically they hung up. Too complicated. As evidenced by the fact within a month, this was all over the internet. An unrefereed, apparently unpublished study in Kerala, India attracted media attention without any scrutiny, posted on the internet. Basically, a rapid drop in the bee population in Kerala, India was the result of recently installed cell phone towers. And that's it. In the meantime, sort of global awareness of CCT rose, and it was clear that there were declines differentially scattered around the globe, with some places even showing increases or no losses at all. And just to be clear, CCD was never really defined. There was no molecular diagnostic. It's a collection of symptoms. It's not really even a disease. May not even be one thing. There was never a molecular mechanism for identifying it. So any decline attributed to CCD isn't really supported by evidence anywhere else other than in the US, yet there was much reference to CCD around the world. And in the meantime, there was sort of a backlash where people in the research community were concerned that there was an overemphasis on honeybees, and that possibly what we weren't seeing as a decline, as these authors, Azen and Harder, suggested, but rather an increase in the demand, and there just weren't enough supplies to meet the demand. And in fact, these authors were invited to write their op-ed for the New York Times, suggesting reports of honeybee decline are greatly exaggerated and that more attention should be focused on native bees. Their analysis was, I thought, compromised by the fact that their population data sort of focused on the period before 2005, which was technically before CCD, which would kind of be like looking at New Orleans economy before Katrina hit. So I actually submitted a letter and it did not get published. So, no $400 for me. More evidence of massive pesticide contamination, 121 different pesticides and metabolites in wax pollen bee bread hive samples, average 6 pesticide detections per sample and a high of 39 per sample. Astonishing levels of contamination. Not much attention on this excellent study, but rather cell phone studies appeared again. This was again from India. They decided to perform a, quote, controlled experiment, placed a couple of cell phones around a hive with a couple of fake ones around another hive. A third had no cell phone at all. Now, a study was published in a journal called Current Science. No statistics, 2 test colonies, 2 experimental colonies. The colony is really the unit of replication, so that's like basically doing a study on 4 people. Current Science, the impact factors it's been mentioned throughout the day. It's a way of estimating how widely cited articles are if they appear in a particular journal. So it belongs to typically the journal over a 2-year period. Nature, for example, has one now about 50. Science, maybe in the 30s or 40s. PNAS is approaching 10. So 0.61 means that over a 2-year period, the average paper in this journal is cited 0.6 times, not even once. Not a real high-powered journal. On October 2010, a new analytical method revealed 2 RNA viruses previously unreported in North America, an invertebrate iridescent virus unknown to science with CCD, and again, the media declared the mystery to be solved. No one in the community thought so. This was so the Congress had mandated annual reports on colony collapse disorder. So special funding. This is the steering committee. Again, I was not an official member of the steering committee. And this was I believe, their final report in 2011. And their conclusion was, although a number of factors continue to be associated with CCD, including all the hypotheses, the reasonable hypotheses from the first meeting: parasites, pathogens, poor nutrition, pesticides, bee management practice, habitat fragmentation, agricultural practice, no single factor or pattern of factors has been proven to be the cause. And then this came back again. This is inclusion of variants with media reports during the year. In May 2011, a new study from a Swiss scientist this time, who concluded cell phone signals can cause bees to make extra noise, a signal to leave the hive. So when cell phones are placed near a hive, it's a barrier. So this refereed paper had nothing to do with colony collapse disorder. It was basically trying to elicit a type of sound communication called piping in bees. And active mobile phone handsets have a dramatic impact on the behavior of bees, inducing this piping signal. So here's your bee. Imagine, this is your house. There are 2 phones with telephone solicitors calling you at regular intervals. It's not at all surprising that these bees were making strange sounds. So. And then we began 2012 with a whole new hypothesis, the zombie apocalypse. Okay, this is a phorid fly. That's a parasitic fly. They lay their eggs on the bodies of honeybees, and then the maggots enter basically the brain and eat the brain, which leads, not surprisingly, to confusion and disorientation. These scientists did not connect this to CCD, but this was a finding, a noteworthy finding. These were known previously in bumblebees, had not been found before in honeybees, but the press embraced this with an embarrassing amount of enthusiasm. As did the public, all kinds of posts. A brain-devouring parasitic fly that infects bees, causing aberrant behavior, disorientation, eventually making them wander away from the hive at night to die, emerging from the corpse a few days later to repeat the cycle. Imagine if that jumped species again into humans. And then the query, will our government attempt to weaponize maggots? And the scientific community was quite distressed by this. Joe DeRisi was involved in the original identification of IAPV, who wrote, it looks like the media is really run with the whole zombie forward thing. Charles and I are authors on that paper. I want you to know we do not agree with the statements being made in the press and by others claiming forwards are even remotely responsible for colony collapse. You may hear from your stakeholders listening to the popular press. Media is way overhyping on this story. And then there are 10 minutes remaining. Well, I guess, is there a sign up yet? Not yet. This was the kind of the last straw of sorts. Chen Sheng Liu is a Harvard professor, but a professor of public health and epidemiology, who published a paper purporting to replicate honeybee colony collapse disorder by feeding bees high fructose corn syrup spiked with 5 minutes? I thought it was 10. 5 till the question? Anyway, I just wasted 2. All right. Bulletin of Insectology, impact factor 0.33. I won't redefine that. Basically, this was a very problematical study. Tiny sample sizes. The unit of replication, again, is the hive. Every hive is different genetically. He used 4 apiaries, 5 hives, 4 different, one hive per each of 4 treatments and a control, 4 different apiaries. That's like doing an epidemiological study on humans in 4 different cities, picking one family in each city and giving each member of the family a different treatment. But literally, that's what it's like. Doses were changed in the middle of the experiment because they weren't dying. And in replicating the symptomatology, No brood remained in any colonies, including controls, but one of the keynote features of colony collapse disorder was that brood was left behind. Also, CCDs characterized by bee death away from the hive. In this experiment, they died in front of their hive. And he speculated, this is not quite reminiscent of the CCD symptoms, bees exiting the hive would have dispersed some distance from the hive if it hadn't been winter. So I don't think you're allowed to do that in scientific papers. It's sort of, if wishes were horses, then beggars would ride hypothesis. And this was roundly criticized. This is a blog from Gwen Pearson. One news item ran with, "Mystery of the disappearing bees solved." No, not even close. I got a bajillion emails about this, about a Harvard professor. And again, all kinds of criticism. Paper's a good example of the problem I mentioned. Some research being given more weight simply because it fits within a narrative of what we believe ought to happen, even if the evidence doesn't support it. Other papers were coming out, much more robust, were getting no press attention at all because they were complicated. He came out, he was undeterred, came out with another study, equally problematical, and he became a hero in the media. There's a very flattering biography, a long piece in the Boston Globe, all kinds of breathless declarations that the problems were solved. John Entine of Genetic Literacy Project sort of did a post-mortem of these two papers, asking people to comment, and it was very difficult because you don't want to criticize your peers, but there was irresponsible stuff going on. And there I am quoted, known for my understated fairness. That's nice. It was really hard to say something nice about this study. It would have been irresponsible to find it. So with my last remaining minute, none of the most likely hypotheses have been ruled out. They all seem to apply. They all seem to interact. SEAL Team 6 ruled out one hypothesis. But a dozen years later, we have interconnecting, interacting factors. That's the best we know, the best we can do. Who's to blame? One study says university press offices. One study says there's a journalistic war on science. Another says it's everybody. Science itself, mass media, science reporting, universities engaging in public relations, self-promotion with varying degrees of literacy. And I just want to finish with a few tips, because I need something inspirational here. Not all science stories have happy endings or endings at all. Please remember that. Don't pick the simpler story over the complicated story to write about because it's easier to tell. It may be simpler for a reason. If it hasn't yet been peer-reviewed, don't write about it yet. If the time between submission and publication of a paper in a peer-reviewed journal is less than a week, odds are good it was not peer-reviewed. Lone genius scientists are vanishingly rare these days, except in the movies. Don't trust scientists who hold their own press conferences. This is something that Chen-Ching Alex Liu did, and who claim to have, quote, unequivocally proven something. And finally, just because you can insert a pun into a science story doesn't mean you should. So I have droned on long enough, as it were. Thank you. So, do we have time for questions? Yes? Do we have questions? No? Oh, I hate when that. Oh, no, that was not one. No questions. Did you know somebody, anybody? Oh, well. Seriously? Oh, thank you so much. Oh. Hi there. Not a plant. I know her, but it's not a plant.
Yeah, I do. I have to give the caveat. I was in Gene Robinson's lab, and I got to watch all of this from the sidelines, and it answers a lot of questions I had about why he wasn't getting very many interviews at that time. I guess I have a half joke question and half serious. I know at that time you kind of moved more into bee research. Was that, how much of that was intellectual curiosity and how much of it was self-defense?
Oh, so the question is, I moved into bee research. Was that curiosity or self-defense? My interest in bees, those honey papers were entirely curiosity-driven. I had been, I know about phytochemicals. Plants make pharmacologically active substances to deter herbivores. They can't run away. They have to defend themselves. And I was, in the mid-90s, offended by the nutrition community publishing studies saying honey is nothing but sugar water. I thought that did a disservice to both the plants producing nectar and the bees making the honey, which is why I did those studies on antioxidant content of monofloral honey. So 14 different monofloral honeys from primarily from one plant source vary 20-fold. And at the high end, like buckwheat, honey has as much antioxidant capacity as like spinach and tomatoes. So it's not just sugar water. It runs these amazing societies, you know. So I was already interested in nectar chemistry and the like. And then, frankly, we found this remarkable reduction in detoxification genes. Then the question became, how do they cope? How do they cope with the hundreds of phytochemicals they encounter? And obviously the 121 pesticides and metabolites. So it was, well, curiosity-driven, and there was funding available. The next slide thanks the USDA, and we're still curiosity-driven. And I have to say, everything I learned about insects up until I started working with bees, I had to forget, because bees are not like any other insects. This is not, I'm not pandering to the bee researchers here. Everything you expect from an insect is upended when you work with bees, because the unit is the colony, it's not the individual. So it was really exciting, actually, to skate on thin ice for a while.
There was a question over there. She's had her hand up. Okay.
Oh, okay.
No, the question that I had was, how do you handle being misquoted or the misinformation by people?
It happens a lot. I was just talking to people at lunch about this. It's easier now than it was. With just print newspapers. I was once quoted by a Southern Illinois newspaper as saying, spiders and other insects. Okay, I'm an entomologist. There are no mind-altering substances that could make me say that. Yet it's in the press, and part of it is, it's the risk you have to take. And as a consequence, I am very careful about what I say. I say it multiple times in an interview, and I ask if I can at least have approval over the quotes. And there's some different feelings among journalists here that's questionable ethics. It's not if the quote is inaccurate. That's fact checking. That's different from twisting and changing things. Because I had one story it was a transcription, a bad quality transcription of by a non-native English speaker who had not been present at the interview, and she thought it was all right to make up phrases that she didn't. That one, I filed a formal complaint with the organization, because it just, again, made me saying things like, it was actually about an award I was going to get, and I was afraid if that went to print, they'd take the award away from me, because it was just nonsense. So it kind of depends on what the origin, the intent and the origin are, and how those quotes are used. I will say, Ed Young is in the audience, and sometimes I can't predict, which one will end up in the story. And sometimes the ones I like aren't the ones that end up getting picked. But that's okay. I'm not a journalist. So. But he gets them right. So, sorry? I'm done. Yeah. Anytime.
This one here. There you go. Is that it? Is that the one you titled it?
Yeah, that's it.
Hello, and thank you, May. Science communication helps us to learn what we don't know, even when we don't know it, and to see what we cannot see. Presenter Juan Perea pursues that mission at the atomic level. Juan is an assistant professor of chemistry and biochemistry at the University of Delaware. He uses supercomputers to simulate the behavior of molecules and atoms. His targets include the hepatitis B and HIV viral capsids, the protein shells that enclose the genetic material of the virus. Those simulations are intended to help researchers learn about the atom-by-atom behavior of large molecular complexes. Please welcome Professor Juan Perillo.
Well, thank you. First, I want to thank the organizer for inviting me and giving me an opportunity to show my research. So it's a very hard act to follow after such a great talk, but I'm trying to do my best to keep you guys engaged with what we do. So we basically use supercomputers to do computational biophysics, but if I tell you that, that sounds extremely boring. So we've coined the term computational microscope. And if we're going to look, I don't know, maybe 1,000 years in the future, this is how we're going to see ourselves. This is a cover figure we made with my wife for a journal cover, and it got accepted. Obviously, it's very pretty. But this is kind of what we do, is like, here are the two of us putting things together to try to understand these relatively simple systems that still have high level of complexity. We combine, because we have the ability to combine everything in a computer, we take data for people who do NMR, you can see your Bruker NMR collection data. We combine things from actually all range of data, from theory all the way to actual observations. So a lot of this work started here in Beckman Institute. I used to work with Professor Klaus Schulten. This was the last picture of the lab. So this was Klaus, here's Klaus. This is a series of figures that were used for a lot of the dissemination of some high-profile papers that we published with Klaus in some high-profile journals. This picture is from November 2013. I tried to look for a more recent picture of Klaus's group, but this is the last picture of Klaus's group. Anyway, so you guys can see what I'm going to be talking here a little bit about is this capsid. Obviously, none of this could have been possible without Klaus's vision, and he really pushes a lot of us to try to communicate our science and disseminate it in the best possible way to reach a broad audience. And we work with Diana in many of these stories. Anyways, so what is this computational microscope? What is it made of? Well, it's really nothing more than silicon. We use very specialized hardware and high level of theory to try to understand how things actually occur in viruses, small organelles, and ultimately we want to understand how cells behave. All of our work is facilitated by state-of-the-art hardware. In this case, this is the Summit supercomputer, which is now the top one most powerful supercomputer in the world. It's still not what is so-called exaflop. It's only 0.2 exaflops. But the idea is that we're going to have an exaflop supercomputer by 2021, and it's going to be in Oregon. So that will definitely facilitate and allows us to study much larger systems and much more complex systems and reach biological relevant timescales. It used to be that all atom molecular dynamics, which is the tool we employ, was limited to very small systems, but because of the combination and the vision of many people, we've been able to push those boundaries far beyond what was originally conceived, which was isolated proteins. I'm going to be talking about these results from two supercomputers. One is this Anton supercomputer that was developed by D. Shaw in New York. He invested a massive amount of his own personal money to develop this machine that actually takes the name of Anton Leeuwenhoek, who was the inventor of the light microscope. So we don't do anything more different than what Anton used to do. We just do a lot of observations. Our measurements as good as the experiment we set up. So we have a fantastic tool, but we're not going to get anything meaningful if we don't set up the right experiment to answer the right question. So let's see how we actually put together a series of these simulations to construct massive structures and to give us a better understanding of the molecular details that are in these relatively simple systems. This is cryo-ET images, cryo-electron tomography images from now world-famous John Briggs. This was before the cryo-EM revolution. And if you guys look at it, these are very low resolution images of a HIV capsid core. So in red is the so-called capsid. You have some other accessory proteins. And in blue, you have what is called the envelope. But at this level of resolution, obviously, you cannot tell nothing more than very basic morphological observations. So we tried to push these limitations and actually build an atomistic model where you have high level of detail, where you can actually study the interactions. And you can actually see things that you cannot see otherwise. For instance, we've established that the morphology of this core that look pretty red, just other than looking like a cone, we couldn't say anything more than that. But now we've established it follows this beautiful arrangement, similar to, it's called a fullerenic cone. And it's primarily made of two types of very simple building blocks, hexagons and pentagons. In this figure, we're showing the hexagons in green and the pentagons in orange. Because we're actually seek to establish truly atomistic interactions, to understand why hexamers are hexamers. And this has very important consequences for many things, including drug development. But more importantly, we are very interested in understanding a series of events that occur in the cytoplasm. And I'm going to get to that later and explain why is that we're so interested in this virus, other than the obvious reason that there is funding and also it's a very important target. This is a cryo-electron microscopy. This was work done by 2013. You can observe that the morphology of the cryo-EM follows this hexagonal pattern. In this part here, I'm going to show you guys what we do with the so-called molecular dynamics flexible fitting, where we use the cryo-EM density to pull the coordinates, to pull the structures into the data provided by the cryo-EM. I'm not going to go into too much detail what these helices mean, but what you need to understand is that now we have a good correlation between the cryo-electron microscopy data and atomistic data that is in this case is represented using this ribbon representation. We actually can do more than the cryo-EM people can do just because of the nature of our tool. So we can also actually, this is the highly controversial pentamer. So when people, experimentalists can isolate the hexamers quite easily, but the pentamer has been, for the longest time, the biggest problem, even with state-of-the-art cryo-electron microscopy technology. So we can actually use our molecular dynamic simulations to establish what are the explicit interactions. In this case, I'm actually showing explicit interactions between residues. In this case, you can get to see the atomic level of resolution. These are tryptophans, for those of you who are familiar with those amino acids. And you can see that we include, the level of description includes hydrogens and this type of thing. What is important to note here, and perhaps you guys missed it in the first run, is that there are these contacts that are broken. And during the course of the simulation, these contacts come together. And this simulation, which we ran for 6 months back in 2012, corresponds like the very first glimpse at an atomistic virus assembly. So it provides some clues in how virus assembly occurs at a very atomistic level. As May was saying, the reason we did the simulation was actually out of curiosity. We wanted to know what will happen if you put a pentamer together with hexamers. And then we decided to run it. It turned out it gave us a very important details that were incorporated into our manuscript. And this is an excellent example of a complicated concept, how to get it across a general audience. So we used this so-called spiral algorithm. So if you think of an algorithm, obviously there is a big piece of code that is associated with this algorithm, but in reality it's very simple to explain. If you actually can look at this spiral, it's nothing more than if you're just essentially counting the pentagons on the spiral and you found the location of those, you can simply by following the spiral. So if you go from 1, 2, 3, 4, 5, 6, 7, 8, 9, and so on, until you have the last hexagon, the last pentagon. So this is trying to communicate a very complex concept, which is a spiral algorithm, which was developed to essentially catalog phyllorenic structures back in 1992. What we do with this, so I don't bore you with the details, is we can go from this one-dimensional representation into a fully embedded three-dimensional structure. So this type of mechanism, this algorithmic procedure allows us to construct these phyllorenic cones following a systematic approach that then they allow us to classify the phyllorenic structures. Something that is very important is the resolution of the cryo-electron tomography is very low. This is an isolated core from actual HIV viruses. And as you can see, there is not a lot of detail that you cannot really tell the location of the pentagons, and you cannot tell the location of the hexagons. So with this, putting this tool together, we actually were able to develop a technique to look at what is the correlation between families of these fillorines and the cross-section, or actually not the cross-section, the embedded capsid, isolated core. So we were able to generate many families. We were able to select what was considered, based on experimental data, a plausible solution for the match between the cryo-electron microscopy data and our embedded fillorines. And we were able to construct a fully atomistic picture of the whole HIV capsid. So in these fully atomistic pictures, we combine what I showed you guys before, which is the structure of the pentamers, the structure of the hexamers. But also in this case, we were more interested in studying the effects that the capsid has on its environment. In this particular case, what is the effect on the solvent surrounding the capsid? So in this case, what I'm showing is the surrounding water that is introduced in the simulation, the location of charged ions like sodium and chloride. And I want to remind you that these are fully atomistic. The representation hides some of the details, blurs the fact that this is a fully atomistic structure that then we can query and perform more experiments. That's what I've been doing for the past 7 years, and I'm going to go into that in detail. So to run these simulations, we took advantage of the largest supercomputers in the world. Obviously, this took almost 2 years to complete the simulation, just because of the performance of the machine and the size of the simulation. So we used for this particular study, there were two supercomputers that were employed. So one was the Titan supercomputer in Oak Ridge, and the other one was the one here in our backyard at NCSA, Blue Waters, which is in its last run. And as I said, we were able to perform, in this case, I'm showing here, like things are moving, they're shaking, and we're actually able to look at the very fine details of the HIV capsid. We made a series of important discoveries. Our first observation was that these hexagons, no one's thought about this before, but obviously hindsight is 20/20. These hexagons actually add as these chloride ions translocation. We saw this permeation through the center of the hexamers, and that was a first for the whole field. We quantified, and we also established a pattern that these ions organize themselves on the surface of a capsid. You can see chloride, which is negatively charged, and sodium, which is positively charged, aggregates on the surface of the structure very differently. So in this case, sodium aggregates on the surface, whereas chloride is located in the interior of the capsid. How can we relate this to the biology of the virus is very important to us. Otherwise, we're just doing physics. We want to study a biological process. We further established that pentamers and hexamers have different binding affinities for these positively charged ions. And we observed this affinity of these sodium ions to these specific regions. And I'm going to get to what we've learned in the past couple of years, that this very first observation, which was simply the ions occupying specific regions in the capsid, that actually has turned out to be proxies for other cellular factors that interact with the substrate. We've performed a very specific, very careful analysis of the physical properties of the capsid. We established a series of discoveries, but I'm going to focus on this particular observation that these hexamers can behave as ion translocation or transport channels for specific small molecules employed by this capsid. Our knowledge of so-called post-entry events in HIV is actually quite scarce. We don't know much about what happens once the capsid cores get into the cytoplasm. So this is our understanding as of 2019. We know this is a virion, which I showed you guys before. We know that the virion goes, there's a fusion event between the virion and the cell that is getting infected. And we know the capsid core makes it into the cytoplasm. There's a series of events that happen in the cytoplasm that are completely unclear to the whole retrovirology field. And here's where we want to make our biggest contribution, is to understand what are the post-entry, the so-called post-entry events. And the main reason we find this virus so interesting is because HIV is an excellent cell biologist. It doesn't matter what you do to it, it always finds another way to infect a cell. This is HIV coming out of a cell. This is, we're gonna get to this later. This is HIV budding from an infected cell. If you guys notice, it has a very different pattern from what I showed before. Essentially looks like these spherical shells. And one of our targets is actually, once the virus is in this immature state, is non-infective. One of our targets is try to prevent the virus from going into this mature state. And I'm gonna get to that towards the end of what I'm telling here. If you guys, I'm gonna focus right now in this little story about our observation that the center of the hexamers can translocate negatively charged ions. In 2016, there came this paper in Nature, and because it's Nature is the gospel, which is not true. A highly controversial study that suggested that the hexamers were used for translocation of DNTPs, or the material prime for synthesizing DNA inside of the cytoplasm. There was they never observed any specific interactions between DNTPs and the capsid. They did some experiments to try to assess what is so-called reverse transcription rate, which effectively what it tells you is the amount of DNA that gets produced inside of the core. However, they never proved that there was any direct interaction, and they furthermore didn't propose a mechanism. So we went ahead and, using our tools, evaluated this hypothesis very carefully. In this case, we actually take a DNTP and effectively study what are the, first of all, energetics in terms of so-called free energies, but also establish what are the steps or what are the limitations of a model where these DNTPs can go through a capsid pore and therefore can be targeted for successful drug development. If you can prevent the DNTPs to going into the cell, into the capsid, you can potentially arrest reverse transcription and have a new type of drug that targets capsid and have a high effect on the life cycle. So again, we use these so-called these simulations, and the only reason we perform this on a massive supercomputer. And I'm gonna try to be a little clear on what we define here is the little scale, which is gonna tell us where is the nucleotide gonna be located along this scale. So if the nucleotide is located in 20, we're gonna have a data point in 20. If we're gonna have a nucleotide is located in 0, we're gonna have a data point in 0, and so on. Similar to any other studies, there is really no hard evidence that these nucleotides actually go through the pore. Furthermore, last year we published in Nature a study that shows that there's another molecule that binds to the center of the pores, which is called mellitic. It's not mellitic acid, it's IP6, inositol hexaphosphate, which goes in completely opposite direction to this nucleotide story. So we have a body of conflicting information. We have some non-so-clear studies, again published in Nature in 2016, that suggest that the DNTPs may be interacting with the capsid. We have very strong evidence from our our work, including crystallography, that IP6 binds to the CA hexamers. If IP6 binds to the same location in this cavity, therefore, that will imply that the channel will be blocked. Therefore, this whole model of the NTP import is completely wrong. However, we still have some reason to believe that the model may be correct based on our observation that the capsid itself has some selectivity against positively or negatively charged ions, as we show in our 2017 study. So to summarize, we have the effect of 3 molecules on the infectivity of the virus. We have this molecule that was used in the 2016 paper, mellitic acid, which seems to have a negative effect on the infectivity of the virus. We have the simple DNTPs, which you guys can notice is nothing more than a sugar and 3 phosphates. And you have this IP6 molecule, which again has sugar puckering and has a lot of negative charge surrounding it. However, it seems that the effect of these 2 molecules, which are seemingly similar, is completely the opposite. And we wanted to answer we really want to establish what's going on using our computational tool, because this is something the experimentalists cannot do. So this is going to be, this is a side view of this hexamer. You guys can see it has, there's a lot of space. This is showing the radius. This is showing here we're showing the electrostatic charge. All you need to know is that it's heavily positively charged at the center of the hexamer. We performed these calculations, and as I said before, this is our location of the nucleotide, and we observed something that is very, very critical, is that the nucleotides can in fact bind, but they're going to bind too strongly. Therefore, once a nucleotide goes into the pore, it's just going to sit there, and it's not going to go through. So the translocation model doesn't seem to be viable with a single nucleotide coming into the pore. We also observe selectivity, so these are so-called potential of mean force. All you have to do is do a subtraction between one point and another to know if it is favorable or not. So if you take a point here and a point here, do the subtraction, that gives you a -6. That means it's a very energetically favorable event. So it means that for 2 types of nucleotides, it's more favorable than the others. But overall, the take-home message from this picture is that you cannot translocate a nucleotide. A single nucleotide will just bind. The other observation is for nucleotides to translocate, they will have to flip. This flipping of the nucleotides, you have the phosphate groups, and this flipping has been observed for other molecules, particularly for aquaporin, which is known to translocate water in cells. However, it doesn't provide a mechanistic view of how the translocation is occurring. So we propose a second model in where there's a cooperative translocation where a nucleotide interacts with an already bound nucleotide, destabilizing the interactions and promoting the translocation. And that has been confirmed looking at these potential of mean force curves, where you can see that the black one, which corresponds to a single nucleotide, has this high binding affinity, which suggests that they just bind, and red one, on the contrary, suggests that things will go inside in the presence of a second DATP, or this cooperative mechanism. But how do we reconcile the picture with the IP6 and the mellitic acid story, which has been troubling us for some time? There is the 2 sit at that top of that little cavity in the channel. However, they seem to have form different type of contacts between those arginines and the molecule in question. So in this case, IP6, because of the sugar puckering, forms less contact with the arginines and therefore leaves the channel semi-open, whereas this is IP6 here, leaves the channel semi-open, whereas mellitic acid lies on top of the channel and prevents translocation, which is consistent with the experiments. And it will suggest that, and show using, again, doing this doing these 2-dimensional potential of mean forces, we showed that IP6, if you guys look at, all you need to know is that blue is more negative than red. In this case, going from the outside to the inside in the presence of IP6 is beneficial. It goes downhill, whereas in the presence of the mulylic acid, you're actually going uphill when you have mulylic acid bound to the capsid. So this provides a very nice comprehensive picture of the effects of these two small molecules, very small molecules, on the translocation model for the NTPs. And our results provide a very atomistic view on an atomistic explanation of the observations performed by the experimentalists. This has allowed us to construct a very delicate, a very specific model of how nucleotides get through the pores, and also this allows us to move forward in the context of drug design, as this is really an important target for drug discovery, as we've already doing that. But as you guys can see, we've identified that nucleotides can go through the arginines. The single nucleotide model is incorrect. If a single nucleotide was going to sit there, a single nucleotide will just interact with the capsid, will just sit there. However, in the presence of mulylic acid, we know that it's a capsid inhibitor, whereas IP6 acts as a capsid enhancer and by enhancing translocation through these hexamers. So this is a very very nice study of how we apply our computational microscopy to understand very complex processes which happen in the cytoplasm. Obviously, we're validating these studies experimentally, and always try to communicate our rather complicated models to the immunologists and virologists, so we can design experiments together to actually test our predictions. We've been working on another series of events that occur post-entry. Another one of those events is how does the capsid actually go from the cell membrane all the way to the nucleus. The RNA HIV is one of those very special viruses that requires its DNA to be integrated into the host genome. So therefore, somehow, it's not just by Brownian diffusion that it's going to go from the cellular membrane all the way to the nucleus. And this has been established that it actually employs microtubules, and it actually uses two types of molecular motors. One is kinesin. It also uses dynein to translocate through the cytoplasm. In this particular case, we're interested in a protein adapter, which is called FES1, which is it's an intermediary. This tube here is microtubule. This one here is kinesin. And then there is this little adapter that it's between kinesin and the capsid, which is called FES1, which actually plays a key role in recognizing the capsid surface and facilitating the transport to the nucleus. There are a series of cells. I mean, these are viruses that infect living cells. So neurons are particularly known to be immune to HIV, and one of the reasons is because FES1 is overly expressed in neurons. So we really hope to understand what are the molecular interactions between FES1 and capsid, with the ultimate goal of developing new therapies in that direction. Again, I'm going to bring back this picture. All we need to understand is that there is a lot of charge inside of these hexagons. And what we've discovered is that the interactions between these small peptide FES1 and the arginines is what actually drives the interactions between kinesin and the capsid, and therefore facilitates the transport, the trafficking of the capsid to the interior, to the nucleus. We've established that, again, this is a very important picture because we've been looking at IP6 for a long time. We've established IP6 interacts with the capsid and is required for infectivity. So therefore, there is an interplay between the two, FES1 and IP6. We've established what the type of interactions those are at a molecular level. What is very special about FES1 is one of those so-called intrinsically disordered proteins, which are very hard to crystallize because of their intrinsically disordered nature, but they're very amenable to simulation, and that's why we've decided to look in that direction. The other part that we've been looking at is we perform this so-called stereomolecular dynamics, in which we apply a force. In this case, we're trying to simulate and try to understand what are the loads that are being applied on the capsid as it gets transported to the nucleus, and if those forces are regulated somehow. In this case, we're showing a protein that is embedded in a membrane, and by applying this force is being pulled out. So typically when you apply these forces, proteins tend to unfold. In the case of the capsid, we actually observe that those interactions are extremely strong. Interactions between phase 1 and the capsid, as you guys can see in this displacement plot versus the simulation time, the whole capsid starts pulling forward, start moving together rather than breaking as the capsid has been designed over the years. So in this model that we have, So in this model, we're actually trying to move this forward towards a realistic binding of kinesin to the morphology. Something that we've learned from building the model is that microtubules have this swirl. That swirl is something that we haven't anticipated and may play a role with the symmetry of the capsid for trafficking. So we're exploring these large interactions between two known large proteins, microtubule and a whole HIV capsid. Again, all of this is facilitated by our use of large computational resources and high level of theory. But again, it's easy to forget that these are atomistic pictures. We actually have very, very, very fine level of detail. We have interactions at the molecular level. We know the organelles of those interactions, and we know what are the effects of the forces of kinesin pulling this massive structure through the cytoplasm. So we're trying to address this question of encoding, which is related to what I just discussed before, trafficking towards the nucleus, and also reverse transcription, which is the biological process associated with the incorporation of the NTPs inside of the core. The last part that I'm going to talk about is this little GAC lattice. As you guys have seen from the previous pictures, we've been looking a lot of cones. In this case, we're going to look at something that is seemingly spherical. GAC is the same lattice that we had before, and it's a very prime target for drug development. So in fact, we have been working with a series of drugs. One of them is in clinical trials, BVM, and it acts by preventing the capsid from going from this spherical form into a mature form. And it does so in a very, very elegant way. And that's something we discovered with employing our computational microscope. So as I said before, then the capsid is made of two domains. You just need to know that there's a green and a red. And there is this little piece here, which is called a spacer peptide. And that spacer peptide was highly, for the longest time, there was a big debate on whether or not it was a helix or not, because people could not see it. And it was primarily because the limitations of cryo-electron microscopy and X-ray crystallography. So there has been a lot of speculation. Back in 2015, we did a study on another virus, a homology study on Rho sarcoma virus, which is a virus that causes cancer in chicken. And the reason we decided to go for RSV is because it's very clear that there is a helix here. For HIV, there was always a debate whether or not there's a helix here, and in particular, the source of the debate is because for another virus, it simply doesn't need that region. You can cleave it, and the virus is still infective. HIV, if you remove that region, the virus is not infective, but it's unclear what is that region doing during the infective cycle. And RSV is very clear that there is something there. So we decided to go in that direction. And it turned out to be the right choice, because I'll tell you why in a second. So back in the day, this is a study we did here in Beckman. This is a chicken virus, and when we went to Klaus, we want to simulate a chicken virus, he was not very happy. But obviously he wanted us to work on HIV, but HIV is this region is extremely problematic, and we don't want to publish bogus science. We want to publish something that we believe in and that we are convinced that is what's going on. So we decided to go ahead and build, what happened? emphasize no signal. Should I get my computer?
Password is. Usually it just goes out.
Oh, really? I can use my computer if you want.
Sorry.
You reckon you know what happened? That's interesting. Yep. Shy. All right, so okay, we're back to this. So people try to build a model of that structure using bioinformatic tools. We decided to give a test to that RSV model that was published in JVI by some of our now collaborators. But the model it was immediately clear that the model was not stable. So as soon as we run the simulation, the model becomes unstable, therefore suggesting that whatever model they built was incorrect. So we actually built a refined model using a series of physical-based techniques, and as you guys see, immediately the model is much more stable. And this was a very important study because it established that, at least for RSV, that helix is plausible that it's there, or established that it's there. But then we went beyond that, just the simulation. Obviously, we identify, again, this is using every single atom in the systems. So we identify a series of contacts in a genetically important region. So we ask our collaborators to do the mutations, essentially a charge reversal that will prevent the formation of these hydrogen bonds. And we can see here in the wild type, as you can see, these little spheres. Those spheres correspond to assembled virus, whereas in the charge reversal, we observe no particle assembly. This immediately proved that, in fact, our model is actually very accurate. It's not my computer. There you go. All right. But this proved that our model is actually quite reliable, but more importantly, gave us confidence that our tool, when properly employed, can provide very valuable results. This work was actually done with one of the Klaus's grad students, Boone. We published a paper back in 2015 of an entire atomistic GAG structure for RSV. This was in collaboration with Klaus. But now, why we never went to HIV is a very different story. So HIV, that region, is extremely complicated because it is an intrinsically disordered region. Unlike RSV, every time we try to simulate the HIV, the SP region for HIV, we never was able to fold. We use massive amounts on the Shaw supercomputer to try to fold it. We do bioinformatics, but we could never get a stable structure. You guys can see here a little bit of the folding pathway for that little region, for the SP region. You can see we did these self-assembly experiments where we tried to go from a completely disordered structure towards a folded structure, and with hopes that we will derive an atomistic model, or at least the kinetics associated with the transitions in this very important region for HIV infectivity. So we were able to fold individual helices. However, we were never able to see an organized lattice like we see for for RSV. We were never able to reproduce the 6-helix bundle for HIV back in the day. And we were very fortunate not to publish anything because last year, I mean, we did publish a series of studies on the dynamics and on the intrinsic disorder behavior of the SP lattice, but we never published any bogus results on the structure of the SP. And in retrospective, it was very wise of us because last year, in collaboration with a group in Cornell, we published a paper in Nature that shows, we established that you actually require IP6 for the formation of the SP bundle in HIV. So we could have forced ourselves to believe that there was such a model in HIV, whereas we were missing a very key component, which is this very small molecule IP6. This IP6 story is a pretty big deal right now in the HIV community. For 20 years, people have been doing these so-called assembly experiments in the absence of IP6. So it shows that for 20 years, we have been doing the wrong experiment in vitro. So now everybody has to reproduce their data in the context of this small molecule, including us. But we were fortunate enough that we never fabricated any data, but we never published anything about the structure of this bundle. However, we have been able to derive a series of small molecules that bind in this same region as IP6. And we did know that these small molecules actually were able to stabilize the 6-helix bundle. So this BVM and a whole series of small molecules actually bind to this important SP region, has a binding affinity higher than IP6 and is competing with IP6 to bind to these so-called 6-helix bundles. And what is very important is there are two effects. The first effect is the ability that IP6 gives the bundle to come together, but also the second effect, and it's the one that we published and we discovered with our long time scale simulations, is that the second part for the virus to be infective is these transitions between helix and coil. The virus is that region in HIV is constantly changing from a helix to a coil, and the reason for that is to be processed by the protease. The conformation of the SP domain must be in this extended conformation. So if you prevent that helix coil transition by binding this overly stabilizing drug, you actually inhibit the maturation of the virus. And we have a whole set of new drugs that are actually quite effective against HIV. They have their own problems right now, but they do so by preventing the helix coil transition. However, IP6 doesn't prevent the helix coil transition. It facilitates that the 6 helix come together and form this stable bundle. So these are also very important results from our computational studies because it established a baseline. Again, it provides a very important picture of the importance of including every single detail, every single atom. When we ignore things that we don't know are there, we get the wrong results, or we get unstable models. So we are always very careful of what we model. We're always very careful of what we publish and how we publish it, because there is always, and I'm pretty sure there's a lot of biologists in here. You know, biology is not like physics. It's always something else that comes out. So this is another figure that we made for a cover for JVI. So we're always making pretty figures for journals. Are pretty successful getting those covers. Ultimately, what we want, as I said at the beginning, we want to go to an optimistic picture of full virion. Full virion is a much more complex system. We are in collaboration with Jody Haddon, who's a carbohydrate expert. We're building these envelope proteins that contain all these complex carbohydrates on the surface. We have a very rich glycosylated model. We have many of these in our envelope. We have a very comprehensive study, interactions of the capsid. We know it's a lot of work I haven't presented today that we've done. We've also established interactions with all our molecular factors, in this case cyclophilin, all with aims at building our mature capsid. Last, we are working very hard on generating a model of GAC, again, including every single atomic detail, IP6, the NTPs, all the type of post-translational modifications that might occur in a realistic, in vivo more than in vitro We're also working with two of the top people in cryo-electron tomography, Florian Schur in Austria and John Briggs in Cambridge. We have these tubes of GAC. We try to use them to generate, validate our parameters. I like these tubes a lot because they're extremely disordered, just like GAC. This is what GAC looks when you simulate it. As you guys can see, it has these massive cracks, unlike the mature HIV capsid, which is completely closed. GAC it has these grain boundary defects that arise from certain physical interactions. Again, as I said, our goal is to build an atomistic model which incorporates both mature and immature. For immature, we have to be very careful, as we've established, with this so-called SP bundle. There's another piece that is extremely concerning to us that we're working very carefully towards, and is the RNA. RNA-NC and nuclear capsid interactions are also very critical. And that's something that we're also working towards very carefully. Again, these are all atomistic studies that require much care. The last piece, perhaps one of the ones we've made more progress, is in building the realistic HIV-1 lipidome. This has been done in collaboration with Tyler already in Los Alamos. We've been able to successfully equilibrate a lipid leaflet. You can see it here being simulated. We have a total of 24 lipid types. HIV is an excellent system because there's it's an excellent system to simulate because there's a wealth of information, a lot of studies over the past 20 years. So the lipidomics are pretty well established. And so we are able to build very accurate, we were able to build a very accurate lipidome. We have 24 lipid types. So we see anomalous diffusion. We're pretty sure we're going to learn a lot of interesting biochemistry with this system. Again, this is the mature capsid. We also have a model for an immature capsid. We're developing a lot of techniques, like trying to overcome technical challenge. Like you guys see, like the appearance of these weird lipid rafts. So trying to detect those, building techniques, building tools to detect those errors and correct them in the simulation. You guys can see this is a fully equilibrated lipidome with no defects. We've developed new techniques, again, to show how we can build these models automatically. It's a huge endeavor to build these models for a single person. So we're moving towards a more automated framework to build these massive structures. We take advantage of a lot of the supercomputers. Our methods have been widely applied to a plethora of viruses, as we have a lot of studies on hepatitis B. We also do studies on RSV and many other retroviruses, just because our tools are generally applicable. And this is another image that we submitted for another journal cover, which is inspired by the work of Andy Warhol. So in brief, we use a computational microscope to really provide atomistic understanding of specific events in the life cycle of many viruses, not just HIV. A lot of our discoveries have resulted in new treatments. Our capsid has yielded a new drug developed by Gilead Sciences, which is now in clinical, it's passed all clinical trials. It's probably going to be commercialized pretty soon. We have our work on SP has also facilitated the development of maturation inhibitors. We ourselves don't develop any drugs, but we help those that develop drugs with a lot of our work. Ultimately, what we want is to have a fully atomistic model, and I noticed this figure is still out in Beckman in the parking lot. So yeah, that's one of our figures. This one is a very pretty figure that we made a long time ago. This one has the glycosylated envelopes. It has a capsid, but obviously we want to go. That one is still at the stage that it's just a picture. We still need to put some more work into it to get it into a physically correct model. All right, last, I just want to acknowledge all the people who have been involved in this work over many years. Obviously, Klaus was acknowledged at the beginning. There's a lot of people who has been collaborating with us, very instrumental. Angela Gronenborn in Pittsburgh, who is the the director of the Pittsburgh Center for HIV Protein Interactions, which we are members of. Obviously, the funding agencies, we get some funding from GBMF, obviously computer time provided by TAC, Pittsburgh, and obviously our center of operations right now is in Newark, Delaware, which is conveniently located close to NIH and NSF, and also New York City for the people who develop drugs. Well, and thank everybody for listening to my little story.
Multiple hands. Who am I going to? When we talk of computational results, everything we have seen here is a lot of data in floating points, and we have kind of manually assigned these colors to make the picture best tell the story we want. And that kind of almost comes post the fact. Now, are there some kind of guidelines that you can give us on how we can make these pictures best represent what we do and not kind of make an error of exclusion, something like that?
So you're concerned that your colors
Because,
might bias the reader?
in a way, yes. Because we pretty much choose these thresholds.
Yeah, so
the way
it's
we
kind of
do.
arbitrary, right?
Yes,
Like when
yes.
I choose the NTD and CTD, I choose them because I know where they are, right? So yeah, I mean, I guess we always choose our colors, yeah, very careful. Like for instance, when you have like a color scale, I tell my students, make sure there's a linear color scale and not a nonlinear color scale to hide details or add details that are not there, right? Which is common. I use these rainbow colors, and those tend to hide a lot of details. In terms of aesthetics, we pick colors using a color palette, just like an interior design person would do. It works pretty well. Yeah. So we just have these Pantone books, and you just use those. And you don't have to even think about it. They just look good when you render them. Yeah. Well.
Hi, my question is how we as like science writers can do better to kind of communicate all of the complication in your images, and like what kinds of questions you would rather have us ask to make sure we're not obscuring any of the details or important details or confusing things.
So in something like HIV, people always want to write about how we're going to cure HIV, and we're very far from that. So I think for me, the hardest is try to tell an exciting story, something that's still exciting, but not oversell it. So the figures themselves, for a lay audience, sometimes don't tell much, right? I mean, obviously, other than the obvious, like spherical and cones. And yeah, so I mean, I think the best interactions I've had with science writers has been not always around the images, but around the story. What we wanted to tell, they can transcribe it to a more general audience.
Did you mention that your significant other knits molecules?
Oh, I didn't mention that. That's her work. Can I show that? Yeah, my fiancée, she knits molecules she makes her own representations by hand. Takes weeks. So she can make a whole pentamer just by knitting it, and a whole hexamer just by knitting it. You know. It's very pretty. I don't have a picture with me, but I know you know it.
So when I know this has happened to me, I want to know how you deal with it when you're dealing with a science writer and there's a story they really want to tell, but it's not quite right. Do you have a good strategy for backing them off of that and getting them sort of closer to the facts?
Well, I always get the draft, and then when you see the draft, then that's when most of those adjustments occur. In my experience, I've never had with the scientific writers, never had an experience where they want to tell another story that is not what we told in the paper. So I've never experienced that. Have you experienced something like that, or okay, a little bit? All right.
Do we have a last question? I have one. Okay, one more.
So you mentioned that you need to communicate your results with immunologists and drug developers. So what difficulties do you experience or techniques do you employ to make sure that your models are accurately understood and employed correctly?
Yeah, that's a very good question. So for instance, for virologists, they communicate with gels, like PCR, right? So if you can design for them an experiment that is going to give you a gel, then that's how I break that barrier. So it's easier for me to speak try to speak their language than trying for them to understand what our models are. One of the biggest challenges working with virologists and immunologists, they don't see structures. They can see, you can show them all you want, the SP domain that is folded. That means nothing for them. All they see is that you did a mutation in some experiment and it gives a different gel. That's all they care. They have a hard problem going through the structural details. So I find it as my job to communicate and help them design together with them an experiment to provide a test for our hypothesis. But it's very hard, yeah.
Thank you, Juan.
Mm-hmm.
We're going to have a 15-minute break. We'll resume at 3:15. So have a break. Thank you.
I'll have to just look.
All right, great.
Yeah, just go ahead and hit restart anyway.
That's what I was gonna do.
Yeah. See if it should sign.
Huh?
When it went blank, what'd you do?
I couldn't do anything. I was
Get
gonna.
it back up.
No, it came up with that screen, but I was trying to get this to just.
Did you just escape out of the PowerPoint or what? Because it wasn't even on there, right?
Oh,
Yes, it was.
It was on there, but not on there?
The problem was I couldn't get back into the computer.
When it went blank, did that go blank too? Or was that up on his, did that have
That came back, it went blank and then it came back up to this.
I feel like, I don't know. I think we should, I don't know. I feel like that HDMI cable's not supposed to be unplugged and replugged that much, like you were saying.
I've been telling him that for freaking 2 years.
Yeah.
We need a more flexible one that's
Or
not so stiff.
Yeah, or some kind of splitter or something that we could at least use.
He brought that up too.
Yeah. See, this room and 1005 are totally Earl's thing, so.
I know. I understand.
Yeah, I know. It's ridiculous. But yeah, I don't dare touch it too much. You know what I mean? Yeah.
There you go. We're trying to get this reset.
Can I upload a slightly updated version of it?
Yeah, as soon as we get this out. Yeah.
Can I also...
You don't have to have a laptop on you, do you?
I do have a laptop, yeah, I can use mine.
Okay. Okay, you might have to do that in case.
I would
Yeah,
actually.
More likely we can get it back.
Yeah, hopefully, yeah. Yeah,
Just wanna
they'll
make sure you
See,
sign it in now. Yeah.
so it did something, but see how it's signing in, and it didn't do that
Yeah.
before.
I mean, so
It just
I-
glitched.
Why don't I can just have my laptop ready. So,
Why don't you just put him on the laptop for now, since he's the last one?
Oh, sure. That sounds good, actually. Is that okay with you?
Yeah, sure.
Yeah, okay. That's easy.
I still need to play the in between.
do I need to get mic'd up too?
Are you guys using the lav mics on him, or?
Yeah,
or-
yeah, if you don't mind.
Yeah, sure.
Yeah, there's a power up here or down underneath there, either way.
Do you have an HDMI?
Yep.
This thing is doing something interesting today.
How long is that?
Pretty long.
Yeah. I didn't know if that goes like into that. Yeah, okay. I think we should at least get it more flexible.
Yeah, that's what we talked about.
Well, yesterday we were talking about an extension. At least that might help.
We can do that, that'd be great.
I'll just go ahead and order one.
Yeah, that'd be good.
I just think he needs to get something more
Like a
flexible.
6-footer or something.
Perfect.
At least,
We'll go up this way.
Okay, great. You have a clicker?
Yeah. Where'd the
It's in the clicker.
Oh, okay.
He put it in for some reason. I saw him do that. Alright, so we'll make sure I don't have any issues with the setup here.
Yeah, see that? It would like together. It's gotta be this
So
cable.
it's the cable.
Yeah.
And I've told him.
I had the clicker. I'd like to change the batteries on that just to be safe.
Alright, let me get you.
Okay, I'm gonna get it in. Oh, we're still, okay, there we go. Yeah, just be careful if you can, because like this cable's coming a little loose sometimes.
Okay,
so-
sure. I'm just going to twist this, and then once it's set, it's set.
It's that, okay. Yeah,
Okay, I can't see this.
yeah, there's definitely something wrong with this. I don't even think it's the connector, I think it's the actual cable.
You know what, do you have a plug-in where we can plug in an adapter?
I don't know.
Any of those work?
Yeah, he does one of the.
Is that one or
This one. Let's try this one. So it's
I got another one here too. That one ain't working right.
I think this will work.
That's not even coming up.
I'm just I turn this off or mute it for the moment.
Oh, he's already got you
Oh, yeah?
muted. Here,
Okay.
let me see it for a second, though. Slide that up so it doesn't shut off.
So do you want it to be duplicate or you want to use?
Presenter view.
Okay.
There's your.
So go extend and then start the slideshow. I don't know what it's doing.
Yeah, can we just leave it to the...
Still live streaming.
Excuse me?
I know, but I got to make
Sorry
sure
about
it's all
I'm
going.
just letting you know, in case you want to,
Sorry about that.
No, doesn't bother me. I think it's probably very entertaining.
Yeah, I'll probably.
Okay, you got it? Okay, I'll send an email to Earl about this and hopefully that'll sorry a little bit. I think you're good to go. Okay.
Hello, everybody. Welcome back. We found some rings. One of our participants found some rings in one of the women's bathrooms, and those rings are going to be at the registration table. So if you're missing some rings, they've been found. This is our last session of the day. Directly after this, we're going to have a book signing. We have 6 titles from 6 different presenters from this 2-day conference, and those are at the table. So if those interest you and you want to maybe get an autograph, this would be the afternoon for that. One of those books is described here by Ed Yong. He's a science journalist who reports for The Atlantic. He's based in Washington, D.C. You might be familiar with this book. If you're a Jeopardy fan, a lot of people are these days, you might know that this book title was turned into a clue. So don't look. For $2,000, under the category Complete the Book Titles, was this answer, in my best Alex Trebek voice, about microbes and quoting Whitman. I contain, in the form of a question, please. What is multitudes? $2,000, right down there. Well, in this book, Ed explores the amazing partnerships between animals and microbes. This book was published in 2016 and became a New York Times bestseller. Ed's work appears on The Atlantic's website and has been featured in the National Geographic, The New Yorker, Wired, Nature, New Scientist, Scientific American, and more. He has won a variety of awards for biomedical reporting. He regularly gives talks and radio interviews. Perhaps you have seen his TED Talk on mind-controlling parasites. Ed cares about accurate and nuanced reporting, clear and vivid storytelling, and social equality. Please welcome science journalist Ed Yong.
Hello. For those who went to Ivan's talk, this is the headline. This is about a clinical trial of a group of bacteria that were meant to duplicate the effects of a fecal transplant, but the trial failed, hence shampoo washes out. So contra me, I'm going to encourage puns. All right, so this is a story about microbes, and I wrote a whole book about stories about microbes. So I'm going to start by telling you one of them. So cast your mind back about 350 years ago and to a town called Delft in the Netherlands, where this man, Anthony van Leeuwenhoek, is about to change the world. So this man is not a scientist, he's not a scholar of any description, but he is a civic servant and a draper, but he does have 2 qualities that are incredibly important for our story today. One of them is that he is insatiably curious about the world around him. He's very keen on exploring that world with whatever tools he can get his hands on. And the second is that he's the maker of one of those tools, which are at the time the most powerful microscopes around. It looks like this. This microscope is sadly upside down and back to front. But if it were the right way around, then at the bottom of this spike, you'll see a little gap. That contains a beautifully spherical lens, which Leeuwenhoek grounds himself and then sandwiches between these 2 brass plates to create an object that looks like this kind of glorified door hinge, but which magnifies objects better than anything else around at the time. And Leeuwenhoek turns this thing onto everything else around at the time, onto bits of animals and plants, and into little samples of water that he collects from the canals and rivers of Delft, from urns and vases that have collected rainwater outside his house. And wherever he looks, he sees these things. Small, animate objects, clearly alive, existing below the threshold of his perception. These are bacteria, microbes. And Leeuwenhoek at this point has just become the first person in the history of all of humanity to see these things with his eyes. And that, to me, is one of the most remarkable moments in all of biology, when we suddenly become aware that there is this whole layer to life that exists below what we can see with our own eyes. And that layer we are now very much aware of. We know of its link to disease. We are increasingly aware of its link to health, about how microbes influence all of these aspects of our biology. Leeuwenhoek wrote to the Royal Society about his discoveries, and by the time he died in 1723, he was certainly one of the Royal Society's most famous and most published members. When he died, he bequeathed upon them this black lacquer cabinet with 26 of his amazing microscopes in them. But for some reason that no one really understands, that cabinet was lost. And still no one knows what happened to it, but the microscopes disappeared. And this was a huge problem at the time because Leeuwenhoek was incredibly cagey about his technique for making those microscopes. He didn't really talk about them very much. So when his gift to the Royal Society was lost, it did leave people in a bit of a bind. It made it very difficult for others to create the same instruments and reproduce his work. Which arguably set the study of microbiology back for a while. Now, when asked why didn't you just tell everyone how to make the bloody microscopes, Leeuwenhoek said this: "Not one man in 1,000 is capable of such study because it needs much time and spending much money, and over and above all, most men are not curious to know, nay, some even make no bones about saying, what does it matter whether we know this or not?" So here is the father of microbiology 350 years ago. He's complaining about money, about public engagement. He has bizarre attitudes about open science. And he doesn't seem to know that women exist. Fortunately, everything is better now. Yeah. So I like telling the story of Leeuwenhoek whenever I do a talk about the microbiome, which is not what this is going to be. But I like telling that story because it has an interesting character who does interesting things. And I think that these kinds of stories matter in science. And I think these kinds of stories are abundant in science. They're all over the place. So those of you who are scientists, these are the stories of your lives. They are stories of triumph and joy, of curiosity and frustration. This is science as it truly is, as a human endeavor done by people who feel things, who have experiences, who are more than just the sum of the papers they publish and the discoveries that they make. But often, this side of science is omitted, dismissed, sometimes even looked down upon as being too fluffy, too hand-wavy, not actually part of the science, and something that that is therefore often omitted in the so-called primary literature, or even in media reports about science. I think that is a problem. I think it's a detriment to the world of science because it disconnects it from this rich humanity that suffuses it. Those kinds of stories are what I relied on when I wrote this book, I Contain Multitudes. Getting people who don't care about microbes, and let's face it, most people don't care about microbes, to read a whole book about this topic is a difficult thing to do. And one very easy way of getting people to care about things that they wouldn't otherwise care about is to get them interested in people who care about those things. And so I Contain Multitudes is not just a textbook about the microbiome, it is also a collection of stories about the people who work in the field and who make the discoveries that allow us to know more about this unseen world, from Leeuwenhoek on to people working today. Now, I stress that not everyone agrees with this approach. So I want to show you all a 1-star Amazon review of my book. Subject cries out for charts, lists, tables, diagrams, footnotes, text. I'm not going to read the whole lot. This person did not like the book, which is fine because a book is a piece of art and art is subjective, and therefore people can totally not like something that I might like. I bring this up, though, because I think that this person dislikes the book for reasons that are dumb, which are the following. Here is what this reviewer believes to be synonymous with science and the conduct of the same and the right, and should be the focus of writing of it. Charts, lists, tables, diagrams, details, complexity, and seriousness. And here is what my reviewer deems to be antithetical to science and the writing of the same. feelings and emotions. God forbid we should ever talk about feelings and emotions when we discuss the science. And to me, this is a very bizarre and retrograde way of looking at science. And I think it dismisses all of that very rich humanity that we talked about. And I think it forces people, it stops people from considering science not only in its best lights as the work of creative, passionate individuals, but also in its worst lights as something that is inherently prone to things like bias and ego and the quest for power or for fame. All of these sides to all of the things that we do as people are very present in the conduct of science. And I think that without telling those stories, we give people a very skewed perception of what science is and, in fact, who does it. I've talked a bit about how powerful it can be to rely on storytelling techniques. I think about this a lot when I think about my audience. So here's an example of my audience. These are just random people sitting on the London Underground. They are bored, they are distractible, they have many other claims on their time and their attention. They may last have done biology at high school. Think about what all of you knew about your respective fields when you were last at high school. They also have this incredible superpower, which allows them to nullify any attempts I have to write something that they might be interested in, which is that they can stop reading at any time. And so everything I do when I construct the pieces that I write is partly about gaining people's attention and keeping it consistently on a sentence-by-sentence, paragraph-by-paragraph basis. I also assume that these people do not give a shit about microbes, because fundamentally, there is no reason for them to do so, okay? And what we might ask, therefore, why do people not care about microbes, or really about anything else that we might want to talk to them about? One common answer, I think, is this, that people don't know anything about these topics, and I think this is the wrong answer. This is an answer fueled by the so-called deficit model of science communication, which treats the audience as if it was some kind of empty vessel into which you pour information and facts and knowledge, and lo and behold, they become interested and maybe they'll change their mind, whatever. This is not how people process information. We know it's not how people process information because this is an empirical line of research that many people have studied, and everything we know about the science of science communication tells us that it's not really just facts or information presented in a vacuum that matter. I think the answer to this question is that people don't feel anything for this topic or for any other that they might not necessarily care about. They have no emotional tether that links them to this material that is unfamiliar to them and not necessarily relevant to their daily lives. And to the extent where people actually do feel anything about microbes, it is that they feel fear and disgust. But you can change that. You can use stories. You can use the stories of people who do the work to replace those feelings with things of, say, curiosity or wonder or just interest. Why do other people care about the stuff that I don't care about is a really fundamental thing to ask about. And because I do stuff like this, because I look for interesting stories of people who do the work, I found that you can make people care in large numbers about things that you wouldn't expect them to care about. Here are 3 pieces that I wrote for The Atlantic that have all massive hits that are about lichens, hagfish slime, and hippo poop, respectively. None of these, if you'd asked me beforehand, I would have suggested to you were going to be massive viral hits, and yet here we are. Lichens was at the top of our website for about 4 days in 2016 over the time of the Republican National Convention. And hagfish slime is still today probably the single most read thing I've ever written for The Atlantic or for anywhere else. So stories can be really powerful. But I think one really crucial thing to remember, which we've heard a lot about today, and which I'm going to talk about for the rest of this talk, is that stories can also powerfully mislead people. And that means those of us who use storytelling and who think about these things have an incredible responsibility to use that power wisely and to think about what stories we're telling, who we're featuring in those stories, and so on. So, for example, who remembers this? Okay, So this is a screen grab of a video that went viral on Twitter at the end of last year, in which a mama bear and a baby bear are climbing up a snowy cliff, and the cub repeatedly slips. So makes it almost its way to the top, slips all the way down to this mountain, comes all the way to the top again. Bizarrely, the mum seems to swipe at it. It slips way far down, finally manages to climb back up, and the two trundle off into the forest somewhere. And this was portrayed as a beautiful tale of persistence. Why can't we all be like the baby bear? Go, baby bear, you overcome those obstacles. Which was exactly what was happening at the end. No, it turns out that this story has a darker twist to it, because it is the story of a reckless drone operator trying to film the two bears and getting too close, causing them to panic, and setting up the circumstances in which this baby repeatedly slips down the slope and almost dies. So what looks like this story of life-affirming triumph and persistence against all odds is actually a story about humans being dicks to animals, which is kind of an evergreen story. Okay, really simple example about what we think we see actually being reflecting a very different narrative than first appears. And this story was at the top of the Atlantic over the midterms, which was interesting. All right, I've written other pieces about areas of science that are nuanced and complex and sometimes problematic in their presentation. So close to home in the area of the microbiome that I've already talked to you about. Much has been said about the effects of things like antibiotics and C-sections on a baby's microbiome. Small pilot studies have been done showing that babies who are born through C-sections or through vaginal delivery end up with different communities of bacteria living on their bodies. Thing is, if you then look at adults who are born via C-section or vaginal delivery, you don't see any differences in their microbiomes anymore. So clearly these things level out at some point in time. And then the question becomes, okay, but do those differences, to the extent that they exist, for the time that they exist, actually make a difference to a baby's eventual health? And the answer is, we don't know, because no one has done that study. But that hasn't stopped a lot of pieces being published about whether C-sections are bad for a baby's microbiome or so on. I wrote this piece about why those stories are much more nuanced and much more preliminary than they seem to be. Similarly, in the microbiome space, there are a bunch of people who have quite recklessly done their own kind of DIY fecal transplants, which, for anyone who doesn't know what a fecal transplant is, it is exactly what it sounds like. You take poop from a supposedly healthy donor and put it in the body of a sick person, except there are several cases in which people who aren't sick take poop from sources that they haven't vetted and done this thing to themselves for questionable motives. And I've written pieces on this as well. This is an op-ed I wrote for the New York Times about how this idea of a healthy microbiome is flawed, that we still don't really understand how these communities affect our health, much less to get to a point when you can recklessly take someone else's microbiome and put it in yours when you don't have any existing health problems and know what is going to happen as a result. To do that and to write a lot of pieces about this practice seems to me to be almost encouraging it, or to give a certain amount of validation to the people who are doing it, which I think is problematic. I've also written a lot of pieces in the fields of neuroscience and psychology about areas of studies that have issues with them. So the one at the top is about oxytocin, the much ballyhooed love hormone or cuddle chemical or moral molecule or other ridiculously alliterative epithets. Oxytocin has a huge amount of hype as this driver of all kinds of social positive behaviors, everything from trust to cooperation to morality. The reality is actually very different. The way it works it certainly seems to influence our social lives in interesting ways, but not in clear-cut and not in entirely positive ways. And I've argued in that piece for Slate that the hype around oxytocin is not only a bad reflection of the underlying science, but also potentially dangerous. The bottom two pieces about two other areas, one about brain training games, in which repetitive simple tasks like memorizing lists of numbers supposedly then generalize to all kinds of other abilities in terms of memory, attention, things that then benefit us in our daily lives. The evidence for that is very weak, as Dan and others have shown in a review. And then also studies on the benefits of bilingualism on executive function. So this catch-all term for all these higher aspects of our cognition are supposedly boosted by being able to speak two languages from an early age. And again, the foundational science behind that has a lot of holes in it for many of the reasons that you've already heard about today. Along similar lines, I've done a lot of reporting since 2012, which was, 7 years ago, on psychology's replication problems, this idea that many classic results in this field cannot seemingly be reproduced by people who are trying to repeat those same experiments, and the reasons for why that might be. All of the things that we have heard about already, things about publication biases, about the ways in which researchers can make choices in their experimental design, flexible choices that almost guarantee them positive results without those results necessarily needing to be true. Now, all of these areas have several things in common. They are large fields of research with seemingly a lot of positive supporting work behind them. They've generated dozens and hundreds of papers, and certainly dozens and hundreds of news stories about those papers. And yet they are subject to these kinds of flaws. Small sample sizes, playing around with experimental design until you get positive results, publication biases. And yet we write about them a lot. And I know this because I used to write about these areas a lot. So when I started science writing in 2006, I did it by starting a blog called Not Exactly Rocket Science, which I wrote on my own and then eventually became part of Discover and National Geographic. This is the blurb that I wrote in 2006 for the blog. And at this point, I didn't ever self-identify as a journalist when I started. That came later. And this blurb, I think, kind of reflects that, this idea that the blog is an attempt to celebrate science, to make it interesting and fun. All the articles in this blog are based on peer-reviewed research and written from primary papers, not press releases or news stories. And that last bit is still more or less good, and what I still try and do. I don't make use of press releases. I write from primary research. But what we see here is the centrality of celebration as a goal. The whole point of this was to be yay, science. And also the canonization of peer review as a metric of quality. What I am saying here in this blurb is that peer review is the marker by which I'm judging research. And I'm treating it almost as gospel, right? As if something appears in the peer-reviewed literature, I'm taking it as being good. And we know, or at least I hope we know, that's not the case. You've already heard many reasons for why that is, why ostensibly bad research gets into the peer-reviewed literature, why a reasonable proportion of that literature ends up being retracted. But this is not the understanding I had of science when I was starting writing about this, which is why two of the earliest pieces that I wrote for this blog, number 4 and 14, were about papers like this. So the fourth piece I wrote for the blog was about a study showing that pictures of eyes in public settings could make people more honest, maybe them behave more pro-socially. 14th piece I ever wrote was about a psychology study about the so-called Lady Macbeth effect, that if you primed people with words that reflect physical cleanliness, that they would also affect their feelings of moral cleanliness. Both of these studies have been discussed as part of this discussion around reproducibility in psychology as sort of flashy effects that that get their way into major journals, that get their way into science writing, but are very difficult to reproduce and maybe don't have the strongest theoretical bases behind them. So I think back on this early work, and I kind of cringe a little bit because it reflects a very naive understanding of how I think science works and what I think good science writing should be. And I feel like I had sort of a come to Jesus moment in, I guess, like 2010 to 2012, as I started realizing more and more about issues of reproducibility. So I started realizing that more and more of the papers that I was writing about turned out eventually to be flawed, that were the subject of rebuttals and retractions. So here's where I am now. Now I absolutely do self-identify as a journalist, and this is what I want to do. So not just to tell good stories, but also to unravel bad ones. So our goal is not to just be a cheerleader or a celebrant for science, but also to hold it to account, to also be a watchdog. This means, crucially, that I am not about outreach. I don't do advocacy for science. I don't even really do science communication. Communicating is part of it, but it is also more than that. And so the underlying premise, the motivation behind the work is different. But also, hopefully, as part of that, comes the discipline of a science journalist, by which I mean I try not to trade in snark or gossip. I'm not out for blood, I'm not out to take anyone down, and I'm certainly not an instrument of vengeance for researchers who feel they've been wronged by a rival or a competitor. It is probably going to be useful before we get further down the road with this to show you this diagram that I wrote a while ago, which is every scientist versus journalist debate ever, in which good journalists write pieces in which they're annoyed with bad scientists, but forget that bad journalists also exist and that many good scientists have had bad experiences with bad journalists, as we've already heard about. And good journalists and good scientists forget that there are bad people on both the respective fields and end up annoyed with each other, while the people who everyone is actually mad at aren't listening because they're not part of the debate because they don't care, they are in the pub. Or, I don't know, you guys probably don't have pubs, the bar, or whatever it is Americans do with their spare time. Gun range, I don't know. So as a journalist, here is what I value. These are the things I care about when I'm writing. Accuracy, above all else. I want things to be right. Being analytical, and I sort of prefer that term to either skeptical or critical. Being thoughtful, like actually thinking about the things that I'm writing about beyond just, is this good or bad? Like, what does this mean? And I'll talk about this more in a bit. And above all else, like really being fair. If I'm criticizing someone, giving them a chance, giving them right of reply. Okay, so how does one do all this? I think that we've heard a lot already about like simple tips and tricks for assessing a study, ways of looking at statistics that might be strong or weak. I think these are all incredibly useful. But I would also remind everyone that there is no easy heuristic for judging the quality of a piece of research, and certainly not when you cover as many areas as I'd like to cover. I write about everything from neuroscience to cancer biology to social science to paleoanthropology. A guide to how to work out whether a psychology study is strong or weak is not really going to help me that much when I'm writing about, say, a new Neanderthal fossil. So you need to have practices that help you work out whether something is strong or weak that work across disciplines. And by far the most important one, I think, as a journalist, is to call people. Like to ask other people who are independent from the thing you're writing about whether it's strong or not. And this is just the central act of what we do all the time, not whether by phone or by email, no matter. It's about trying to seek outside expertise, outside independent expertise, and then integrating across all of those opinions to show, to essentially do a form of peer review. And this is also why I disagree with this idea that you should, that science writers should only talk about peer-reviewed science. I write about preprints. I write about preprints quite often, actually, and I think that that is a good practice for science writing. And I think it works because I don't deify peer review anymore. Like the fact that something is peer-reviewed is fine. The fact that something isn't, is it going to be at some point? Is the plan to submit it to peer review, but you're putting it out there now? In which case, fine, I'm going to do the job myself, and I'm going to send it out to people who I think can give me an opinion about whether something is strong or weak. And I think part of this, the really important part of this is to pick your people wisely. So if you're writing about something that's interdisciplinary, for example, you need to get opinions from people across those disciplines. If you're writing about something that is statistically complicated, you need to call a statistician, and so on. If you're writing about something that's ethically complicated, which I'll get to later, you need to call an ethicist. All right, when I do this, what I'm really after are these two things, critique and context. So critique, I want to know whether the thing I'm writing about is solid or not. The method's appropriate. Are the conclusions valid? Tell me if I'm about to do a long piece for The Atlantic about something that's actually bad. I do stress that it is really, really important to people to, if you do have criticism, to be honest about it. I really appreciate when people are that. I do recognize that some people are loath to criticize their colleagues in public, and that certain groups of scientists might face harsher repercussions than others for being critical. If that is the case, then at the very least, let me know off the record or on background so that I have some sense of what weaknesses of a thing I'm writing about might be. Context is just as important. So whether a new thing exists in the background of lots of other things that are similar, or a bunch of historical research that I need to know about to write about something properly. And that's really important because there are loads of ways of writing bad journalism beyond just writing about weak studies. So here's an example. I wrote this piece about a new computational platform that, to allow people who study infectious diseases to analyze the results of sequencing runs and to very quickly work out what's in their sample. There's nothing about this piece that's wrong or inaccurate, but when it was published, it became very clear to me that from the critiques I had of it, that this is just one of many different computational platforms that basically do the same thing, that are largely unmentioned in this piece, which more or less portrays this new thing as like the only thing or some big significant breakthrough, when it is in fact like one of many. And I don't like doing stuff like that because I feel that I have failed my readers in properly grounding anything that I'm writing about in the context of what else is going on around it. So, lessons learned from stuff like that. I had hints of that when I was doing the reporting and got comments from other people, and probably I should just have listened to my instincts on it, because it is totally okay to kill stories, right? Like that is almost the most powerful thing we can do, to just say, no, I think like based on what I found, this is not worth reporting. And you can do that for many reasons beyond just this is bad. Sometimes things are bad in interesting ways, and that makes it worth writing about. But not being able to do your job properly is one good reason. So here, these headlines, none of which were by me, were all about the announcement that Ebola had been discovered in a Liberian bat, which was the first time that virus had been found in West Africa, the source of the largest Ebola outbreak of all time, and that this was important because the exact reservoir of Ebola is still largely uncertain. But this came through a press release that was announced by the Liberian government, and a very credible group of scientists, there was no paper, there were no publicly available data sets. There was nothing beyond the fact that these people were claiming to have done what they had done for me to even show to other people to say, does this check out or not? And so I ended up not writing about this because fundamentally I can't do my job if I can't run something past an independent group of people to work out whether, to get that critique and that context. Okay, so I ended up not doing this. One really important point I want to make for the next section is that we have talked a bit today about good science coverage and bad science coverage and strong science and weak science. I would encourage everyone to not dichotomize things that starkly. Things are not always just good or bad or strong and weak. Everything contains multitudes. I have regrets now. And so in that gray zone, there are tons of interesting stories to tell. So here's one. This story is based on 2 preprints by the same group of researchers, one showing that plants can essentially hear, are sensitive to the sounds of pollinators, and another that plants produce noises their own. So both aspects of auditory communication are there in the world of plants. A really interesting thing happened when I sent this out to a bunch of, I think it was like 6 different researchers who studied work in this area, some of whom I expected to be very skeptical about all this, and some of whom I thought might be more sympathetic. Regardless of that breakdown in my head, every single one of them loved one of those studies and hated the other one, which is really interesting because they come from the same group of researchers. And there is gold there. I think talking about why those 2 studies got very different responses from different people, regardless of their pre-existing conceptions, is a fascinating tale of science and how it works and how it gets analyzed. Anyone seen Our Planet? Okay, for those who haven't, sorry, spoilers. Okay. There is a scene in Our Planet in which some walruses in Russia climb to the top of cliffs and fall off to their deaths. And I wrote about this, having spoken to a bunch of people who worked on the sequence and some other walrus biologists. And Dan makes some really interesting points earlier about causality, and the path of causality here is really interesting. What the show is claiming is that climate change is leading to lower sea ice levels, which is pushing walruses out onto haul-out beaches in larger and larger numbers, and that overcrowding is causing some of them to climb up the cliffs, and climbing up the cliffs is sending some of them to fall to their doom. There are many steps in that, and some of the walrus biologists I spoke to were really happy with the first elements, but not really the others. And so this whole claim of is climate change causing walruses to fall off cliffs is not necessarily wrong, but it's complicated. And so unpacking that and looking at how people are understanding different parts of that causal chain is interesting to me. This is a piece I wrote about recent claims about the insect apocalypse. I interviewed May for this. I hope I quoted her fairly. So you may remember a slew of claims a few months ago about how insects were dying in such numbers that maybe insects might disappear altogether within a century. I think that claim is ludicrous, but there is some interesting stuff here about how not just the extent to which insects are disappearing, but also how we make sense of imperfect data. So, and May can correct me if I'm wrong about this, but my understanding here is that there aren't that many good long-term data sets, and they cover a small proportion of the total insect fauna that they are, but those data sets we do have are pointing in an alarming direction. So given that we don't have enough to make representative, super generalizable claims, how do we react to the piecemeal data that we have in a situation where we're actually talking about the difference between not just life and death, but existence and extinction? Again, an interesting way to look at this beyond just, is this right or wrong? Similarly, there was a really interesting debate that came on the back of a science paper that looked at how much of the ocean is actually fished. The original paper said it was 55%. The critics analyzed the exact same data set and came to 4%. And they do that because they're taking very different looks at exactly the same data and ways of measuring fishing impact. And they both have very reasonable claims for why their particular metric and their way of measurement is the right way. And so this piece presents that. And I think, again, the interesting question here is not actually what is the number or like, was the original paper bad or not, but how do we make sense of the world? How do we take a question like this and turn it into an analysis plan? How do we get numbers to answer this question when it's actually not that straightforward? I think about this a lot because our editor-in-chief, Jeff, often says that people don't come to The Atlantic to learn about the news. They come to learn how to think about the news. And that's sort of what I meant when I talked about thoughtfulness being a value that I aspire to, that you're not only telling people what is happening, but you're hopefully giving readers tools to think about the world beyond just like yes or no, right or wrong. Like ways of thinking about and analyzing the news that they're hearing within the pieces that you yourself are writing. So I also am interested in things that take a hard look at entire fields of science and ask whether they're going in the right direction or not, beyond just are things reproducible or is this study right? So here is a piece that I wrote about neuroscience, about which argues that many recent studies, so this recent push to prioritize flashy new tools, often leads us to forget that brains exist in animals and that those animals behave in interesting ways, and that behavior is a really crucial part of understanding how neuroscience works beyond things that allow us to, for example, track brain activity at a network level or to piece together the firing of a single neuron. This is a piece I wrote about attempts to resynthesize an extinct virus called horsepox, and the ethical question of whether this was worth doing in the first place or not. Again, this is not just about is this ethical or not, it's also about the fact that the team that did the work got to almost unilaterally make the decision about whether to do it or not. And that's interesting to me. That reveals a flaw in the way modern science is done, in which a lot of research goes on without people knowing about it. And you only know about it after the fact, and you only get to have these ethical decisions after stories have come out, papers have come out, news pieces have come out. Is that a thing that we need to worry about? This is a piece about cancer biology, about how the media that scientists have been using to grow cancer cells for many years, while very easy to make up, doesn't actually reflect the physiology of the liquids in which cancer cells truly exist in our bodies. Is that biasing research in important ways? And finally, there are just areas which are just outright ludicrous, like the announcement late last year in which He Jiankui told the world that he had allegedly created the world's first gene-edited infants, in which a gene that was supposedly, in which he had edited a gene using CRISPR to supposedly make them more resistant to HIV. This has so many problems, scientifically, technically, ethically, in all sorts of dimensions, that I was forced to write a fucking listicle. So, stories matter. Again, I definitely think they matter in terms of talking about science, in terms of showing science as this product of human endeavor. But the story is not just science is great, it's also that science has a dark side. And I know I'm nearly out of time, but you're going to have to bear with me for 5 minutes because this next bit is important. Thank you. All right, so the last thing I want to talk about is who gets to be in our stories in the first place. And a lot of my thinking about this was inspired by and influenced by a colleague named Adrienne Lafrance, who now runs basically The Atlantic, certainly our website and our podcast. She's one of the best journalists I know. And a while back, she did an analysis of her own stories in which she looked at the gender split among her sources for about a year. And she found that only 25% of the people whom she was quoting were women. This proportion just seems to be like a general media thing. Like whenever anyone across different publications has done this type of analysis, they typically find something like 25%. So 3-to-1 overrepresentation of men to women. And what Adrian wrote at the time was that by failing to quote or mention very many women, I'm one of the forces actively contributing to a world in which women's skills and accomplishments are undermined or ignored and women are excluded. And that world is very much in our faces all the time. It's certainly very much in the face of anyone who works in STEM disciplines. So women face stereotypes about their scientific acumen. They are deemed as less employable or less competent than their male peers. They're paid less. They're invited to give fewer talks. They offer less mentoring. They're subject to ridiculous amounts of harassment and abuse. And and to the extent that any of that applies, it all applies doubly to women of color. I always fancied myself as someone who cares about this, so I was very surprised when I did the same analysis as Adrian and found that I also had the same ratios, 25%, 3-to-1. And this felt just unacceptable. Felt like I was contributing to the same world that Adrian was describing, and that as a journalist, as someone who works in the media, I am essentially a gatekeeper. My choices of who I feature in my stories influence who my readers see as a scientist, what they think a scientist looks like. And as we heard from May's talk, those decisions about putting people as voices of science have huge impacts often on their subsequent opportunities as spokespeople, as professionals in their own right. So I've been trying to fix this ever since, so I keep a spreadsheet now where I do a running tally of all the stories that I write and the proportion of sources who I contact and quote in my stories who are men and women, who are people of color. That ratio of 25% only took about 4 months to raise to about 50%, where it has stayed ever since. I'm also doing the same, like I said, I'm also doing this analysis for people of color too, who currently stand at about 27% of the people in my stories, which is not bad, but also lower than I would like. We've talked a lot about good or bad science, about statistically strong and and statistically weak science. To me, a science that is not diverse, that is not inclusive, that doesn't care about the representation and the opportunities and the lives of the people who do that work is also a bad science. And I think we shouldn't tolerate it any more than we should tolerate irreproducible work or bad, inaccurate stories. And I think it's important because the people who are in science shape the science that gets done. I think that should be fairly obvious. Again, this just goes beyond gender. This is a story I wrote about an ancient DNA studies about the peopling of the Americas, in which I really tried to make sure that I got voices from indigenous researchers, including indigenous geneticists, to see how their perspectives shape their views on this work. This is a study I wrote about young gender non-conforming children and the decisions that ultimately go into whether they transition or not. This piece quotes several transgender scholars, including at least one transgender scholar of color who is an expert on statistics herself, perfect source. And I've been fortunate to have a lot of people I respect whose work has informed this thinking. I actually wrote a piece once about this person, Ben Barres, who was a Stanford neuroscientist. He passed away, sadly, a few years ago. Ben and his colleagues and proteges were instrumental in showing that the glia cells, the cells that make up about 50% of the brain, not neurons, are and that have long been relegated to being the sort of architectural supporting players in the brain, are actually really, really important for all these kinds of aspects of neurobiology that we had underestimated them for. Ben also spent a lot of time talking in all kinds of talks, even his just basic neuroscience talks, about issues of diversity, challenges faced by women in science. And he knew those challenges well because he was a transgender neuroscientist, so he very much understood the challenges of people at different ends of the gender spectrum. I think about Ben a lot because I don't think it is any coincidence that someone who thought very hard about the problems faced by marginalized groups in science and issues of diversity was also a person who eventually showed that a group of cells that make up half the brain and had long been overlooked and unappreciated were actually super important. Again, I think the people who we are and the backgrounds that we have shape the scientists who we become and the work that the questions we ask about the world, the way we interpret the results of the answers to those questions. And I think that's important. That's why I am trying to make an effort, an ongoing and growing effort to prioritize this in my own work. I think a lot about the work of my partner, Liz Neely, who runs an organization called Story Collider. Story Collider gets people, scientists and non-scientists, to tell true personal stories about science in live stage events. They have made a priority of getting people from different genders, sexual identities, racial groups, people with disability, mental health issues, all people from all kinds of different communities and groups onto their stage to tell their stories, to share the full force of their person on stage. And there was a really interesting small study at a community college where someone got a bunch of students to listen to these stories over a course of a semester and qualitatively looked at how that changed their perception of what science was and how they saw themselves within it. And many people talked about how they felt a place for themselves in science. They could see themselves reflected in science through the identities of these storytellers. Again, this is why I think that this work is incredibly important and why I think we should prioritize it just as much as we prioritize being accurate and fair and analytical and all the rest. Thank you very much. Yeah, for sure. Hi.
So how do you find your science sources, and has the process become any more difficult when you try to bring minorities and women up to a level of?
Yeah, so that's a good question. So I basically just do the same process I use to find sources, but spend more time on it. And that could be like just doing like a simple Google search, like professor and then like the topic that I'm interested in. I'll try and do searches for papers, look at the authors. I'll look on Twitter, see who's talking about a specific thing. There are some databases for sources from diverse groups. 500 Women Scientists runs one that's pretty good. Sometimes those work, sometimes those don't. A bunch of these different techniques almost always yields people. Now, it does take time. It's not a non-zero amount of effort, but it takes much less time than I think people might think. And to me, it's well worth doing. Often I found, like in rare cases when I'm writing about a very specific thing and need a very specific amount of expertise, sometimes there just aren't people of color. Usually there's women. Some very, very, very rare cases there are not, which is fine. But often, more often than that, I will be writing about areas that I have covered a lot that I will count as my beats, and discover people who I've never heard of before, which then begs the question, why have I not heard of them? You know, like what systematic biases existed in the world that I, who supposedly am meant to know this area really well, had no clue that this person even existed? And this is why I think doing this work is important. And also, like, it does get a little easier, but I also try and continually make it challenging. Like, I think there is a problem when, if you're writing about a specific area regularly, to repeatedly just go back to the same person you find there and then, because like usually people from marginalized communities have a lot of shit to deal with. And they're already like burdened with all kinds of things that the people from more privileged groups are not. And so to me, to repeatedly go back like every month saying, what do you think about this paper? What do you think about this paper? What do you think about this paper? Is probably not helpful to them. So every time I write about a new thing in an area that I cover regularly, I always try and find someone new. And there's always someone new. Hi.
Could you hold your question till we get the remote to you so that we can get the question for remote people? Microphone to you.
Hi, I appreciate your perspective on this a lot. I'm a faculty in mechanical engineering, obviously a woman. I wonder if you get pushback on this, and if so, how do you handle that?
I wouldn't say I've had ever any like credible pushback on this. That piece that I put up about I spent 2 years trying to, about like 2/3 of the way down, I actually try and preemptively address the most common yeah buts, of which by far the most common is, shouldn't you be thinking about quality? Like, I am thinking about quality. I'm getting people who are experts in their fields. It just so happens that now half of those experts are women, and some of them, and a quarter of them are people of color. It is interesting to me that this question of why don't you look at quality never comes up in the status quo when I'm quoting 3 times more men than women. Like, why aren't we asking questions about whether those people are the most qualified people and all the social and systemic factors that lead to them being the most obvious and prominent candidates while not necessarily being the best ones? So yeah, there is pushback. I would say it has been surprisingly limited. And I think, like, which is again, I think why like it's important for men to talk about this, because we're probably less likely to get pushback on this than a woman making exactly the same point, and that is our privilege and something that we can use to raise awareness of this issue.
Thank you.
Thanks. Anyone else? Hi.
I really appreciated what you had to say about using stories to talk about science, but one of the problems that I've had with that as a scientist is that we often fight against the use of anecdotes to disprove science. For example, anti-vaxxers often use many anecdotes to try to prove their point. And how do you differentiate that and make sure that your stories aren't overshadowing the science that you're really trying to communicate?
Okay, so one really crucial thing I want to say is that I don't think that those two things are separable, right? So when we talk about the stories overshadowing the science, I think implicit in that is the idea that those two things are disparate, right? And that the stories are just a tool that you might use to tell people about the science. I think the stories are the science. I think that the lives and experiences of those people who do research are inextricable from the work that they've actually done. And I think that you can, but I think that it is counterproductive to treat those as things that you can dissociate, and for one to just be a tool and the other to be the real stuff. Now, I do get that it is problematic that people use single anecdotes as like counter-arguments to entire lines of research. But I will say that like these things are powerful, right? This reflects how people think about the world. Like one person's story about their child and their experiences as a mother trying to care for that child is more powerful than a meta-analysis or an n equals however 7-digit study, right? Like we can do two things. We can rail against that and despair about it, or we can understand that in understanding that is in itself an evidence-based proposition. Like when we think about communication, we're not just thinking about the things we're trying to get across, but the ways in which we're telling people. And the ways in which we're telling people is an entire field. Like, how best to deliver a message is an empirical question to which there has been research. And that research tells us that things like anecdotes are powerful, that stories are powerful, that appealing to people's identities, their values, their experiences is powerful. So we can either, like, I actually think we should use that. I think we should respect it, and we should respect the power of using it badly, and the risk of doing that. But I don't think you can just say that, like, I think one of the flaws of many approaches to science communication has been to think that we're better than that, or to sort of dissociate ourselves from it, whereas in fact, if we actually think about how people process information in other parts of their lives, we all do this. We just need to do it well. Okay. Hi.
I want to follow up on that because I'm a scientist turned journalist, and I do feel compelled to make sure that, you know, I recognize the power of anecdotes, but I do feel compelled to make sure that the anecdote I'm using is kind of representative of a thing, that it's not like an outlier.
Right.
So, I mean, I know you agree with that, but I was wondering if you have a method for how you kind of assess that when you're checking that the anecdote is representative.
I'm not sure. I tend not to, like, I try not to offer, like, strong heuristics for these types of things, because I think that, like, they vary so much on a case-by-case basis. And actually, like, I think sometimes we forget, because the most prominent examples of debates and controversies that we deal with regularly, like evolution or like climate change or anti-vaccination, are very stark, right? They have an obvious answer to us. There is consensus. That's very much in the minority, right? Like 99% of controversies in science have no such thing. There's still areas of like active debate. So like actually finding a like, this idea of finding a representative anecdote like does kind of hinge on there actually being a right answer and on you knowing it. And in many cases, that's not the case. So I think like it's about trying, like, again, I go back to those values of like being analytical, being thoughtful, being fair, like looking at the perspectives you're putting on a page. Like if you're dealing with an area where there are legitimately two sides, like are you presenting one above another? Like if you're it's an area with consensus, like are you presenting the opposite? Are you now presenting those things unfairly? Like it's a very, very fine judgment, and it's one that you really do need to make on a case-by-case basis.
One more question?
Let's take at the front. Hi.
Hi, so I'm a public information officer at an interdisciplinary research institute on campus, and they talk a lot about team science, and I like to think about team communications. And so when you said that you don't make use of press releases, I was a little bit surprised by that, and I wondered if you could clarify how you view the science communications sort of like ecosystem.
Yeah. I mean, it's tough for me to say because I don't make use of press releases. Like I prefer to go to to primary papers. I like it when scientists reach out to me directly, and I find ideas by going to conferences, by looking at debates online, by doing all those sorts of things. Like press releases, by and large, aren't that useful to me. Now, they are useful to a lot of people, so I'm not doing the down or disrespecting the craft of doing a good one. It's just not something that I specifically rely on.
Are scientists connecting you with PIOs to get additional tools to help you tell your stories? Like, are you getting photos or videos or this additional content? Or are you even getting that through your individual reporting?
So often the latter. I mean, videos are not, unless they're like very specifically about the thing that I'm writing about, they're usually not that useful. Images I'll ask researchers for themselves. I like having that direct line of contact. I honestly think it makes for better journalism.
Okay, thanks.
Thanks.
Thank you, Ed. We have the book signing now out in just outside the auditorium. Breakfast in the mornings at 7:30. We're going to start our first session at 8:25. Have a good night.